Good morning, ladies and gentlemen. Welcome to our final day of the International Symposium. We have another very, very interesting thematic session on our program this morning, and I'm relieved to not having to chair this because we have two really very competent chairs and moderators. First of all, Beverly Glover from the University of Cambridge. She's the chair of the HFSPO Council of Scientists, together with Peter Langridge from the Australian Academy of Science. Beverly, to you.
Good morning, everyone. So this is a really exciting and important session to be part of. We talked a little bit about this at the summit on Tuesday and agreed that food security is one of those problems that is particularly challenging in a changing world, because the challenge was there already, how to feed everybody. The challenge is made worse by climate change, by the physical effects that has on habitats and the places crops grow, and also the effects it has on human communities and how they move and migrate. And also many of the apparently obvious solutions to the problem will only make matters worse by deforestation, habitat loss, and more loss of biodiversity. But it's also one of those problems where an interdisciplinary approach is really beginning to produce hope, because we start to see solutions emerging from different technologies and from the combinations of different technologies. And this morning we have a really exciting session because we're going to hear about several different examples of that hope from our great speakers this morning. So I'm going to kick off straight away by introducing Gabriella Schlau-Cohen from the Massachusetts Institute of Technology, who's going to tell us about some of her work on why plants don't get sunburn and how we can use those ideas to help us harness sunlight.
Great. Well, thank you very much for the introduction. Also, thanks for the invitation. It's really an honor to kick off the third day of what's been an instructive and inspiring summit and symposium. So thanks to HFSP and all the staff for putting this together. So you all know from life that unlike you and I, plants don't slather on SPF 50 when they go outside on a sunny day. Yet, except in a few limiting cases, they don't get burned. Now, the reason for that is because they've evolved their own version of sunscreen. One of the major focuses in my lab is understanding the molecular mechanisms behind this sunscreen. The reason that we're so interested in this process is both to advance fundamental knowledge and because of what's shown here. So I showed this 2 days ago, but I think it bears repeating. What we see here is for 4 major cash crops, yield as a function of time, with predicted demand shown in dotted lines and predicted supply in dashed ones. And we see this growing mismatch between supply and demand. And so one of the biggest challenges facing us as a society, as Beverly laid out, is figuring out how to bridge this gap. One way to do it is to increase the supply. So fundamentally, the supply comes from photosynthesis. But we know from our fundamental studies of photosynthesis that it operates at this measly 1 to 5% power conversion efficiency, meaning just this small percentage of energy gets used to generate the biomass of the plant, including all the food that we eat. And we know that the rest of the energy goes to a range of different processes: carbohydrate synthesis and inefficiencies in that reaction, the metabolic processes like photorespiration, and environmental responses, which I've drawn as the biggest slice of this pie here, which is, for example, the conversion of absorbed sunlight to heat. But the size of these slices changes with factors like nutrients and weather. And so if we really want to understand what controls the size of these slices and begin to think about ways in which we can change them, we need to look at molecular mechanism. So looking at a crop field like this one, we want to zoom in on the molecular machinery. And we find incredibly complex nanomachinery. The protein shown here, I introduced briefly at the summit, but it's protein near and dear to my heart. It's called light harvesting complex 2. This is the protein that makes leaves green. It's also the most abundant membrane protein on Earth. And so this protein is responsible for capturing sunlight and in part controlling the partitioning of that sunlight between the different slices of this pie. So if we want to understand these slices, and today I'm going to focus on one of them, thinking about how absorbed sunlight generates heat, one of the things that we know is that this level of heat can change dramatically, like I laid out at the beginning. For example, with weather. So I showed it originally at 70% of absorbed energy gets converted to heat, and that can certainly happen on a sunny day. But under clouds, it can be less, taking on a range of different levels. So if we want to think about optimizing the amplitude of this slice of pie, what we need to think about is what are the molecular mechanisms at play in this protein here. So if we want to think about the molecular mechanisms of dissipation, we can start by thinking about why is it that dissipation is happening at all? Why is it that these proteins have evolved processes to take sunlight and convert it to heat? And that's actually shown in this plot right here. This shows sunlight on a typical day at a typical point on the Earth's surface. And what we can see is a profile of sunrise, sunset, along with clouds and shadows. Now, sunlight up until around this level is useful. This matches the turnover rate of the molecular machinery of photosynthesis. But typically, energy in excess of this is dangerous. A little more rigorously, what happens is the chlorophyll molecules involved in photosynthesis absorb sunlight, undergo what's called an intersystem crossing to generate what's known as a triplet state that can sensitize singlet oxygen or reactive oxygen species. And these reactive oxygen species, as all the biologists in the room know, damage cellular machinery. In fact, they're why I hope you all eat fruits and vegetables, because these reactive oxygen species or free radicals can damage our own machinery, and we need the photoprotective mechanisms of plants to eat them to keep them safe. So this is what they've evolved to do in plants. We can't do the same thing, so we eat our fruits and vegetables to make sure. All right, so getting back to this, so ideally, plants would dissipate the excess energy, retaining only the useful, but all of the useful energy. Now, the problem is that plants are cautious. They keep much less than this ideal level. And so if we want to think about this mechanism, what we fundamentally need to understand is how is it that this level of dissipation is controlled? So this is a regulatory process, which means we can conceive of it as a control loop, where we have our input, our desired output, and then the control loop that converts that input into the output and sets that level of that dissipation process. And so the fundamental question, if we want to think about this slice of pie, is what are the molecular mechanisms behind this control loop? And so what we know is that energy gets regulated via a protein conformational switch. So proteins, like light harvesting complex 2, have embedded within them two types of pigments: chlorophyll, the primary pigment of photosynthesis, and carotenoids, which serve a variety of roles, including as the free radical scavengers that we need to eat. And so it's these chlorophyll and carotenoid molecules that capture and manage solar energy. So they can absorb sunlight and use it to direct it towards the generation of biomass, or they can redirect that energy into heat. So we want to understand the conformational or structural switch of this protein that goes between these two states. So one of the mechanistic questions about dissipation is, what is the conformational switch that turns it on? And so this is the mechanistic question that I'm going to talk to you about today, as an example of the kind of work that we do in my lab to understand dissipation in plants. Okay, so what's been known for decades from a range of molecular biology and biochemistry studies is that the level of dissipation gets activated by two factors: by a drop in pH, which means there's accumulation of protons, and a change in the chemical composition of one of the carotenoids, where it gets converted through an enzyme from violazanthin to zeaxanthin through a deepoxidase reaction. And I'll just say that that activity of the enzyme is in turn controlled by pH. So it's known that the dissipation, or the conformational switch, gets activated by these two factors, but what changes in this protein had been a long-standing question. And one challenge is, if we want to understand the conformation of the protein, typically when we do protein-based experiments, biochemical or spectroscopic, we might be measuring around 10 to the 4th individual proteins. So each one of those proteins will have the switch toggled to a different state, meaning it's in a different conformation, or undergoing a different part of the dynamical process. And so typically what happens is measurements average over all of these things. And so one of the things that we do in my lab is we take these proteins and we put them in a really sensitive microscope, so we can look at just one protein at a time. We can identify its conformation to determine the state of the switch. Now, what we actually measure in these experiments is what's called the fluorescence lifetime of the protein: how long it takes sunlight or solar energy to come back out. This tells us about the level of dissipation. If energy is only coming out for a short time, that means there's more dissipation. And so this gives us a way to determine the state of dissipation in individual proteins. From measuring many of these proteins, we can figure out what is the state of the switch under different conditions, putting together a picture that's known as a free energy landscape, which fundamentally tells us about the relative populations of different protein conformations under different conditions. And this gives us a window into the level of dissipation and how it changes. Along with this, we can look at the dynamics to figure out how this process responds to different changes in sunlight. So we've taken this approach and looked at a range of different photosynthetic organisms. We've looked at proteins from green plants, and remarkably, we didn't really see any dynamics. Tells us that the plants get fixed, or these proteins in the plants get fixed in a certain level of dissipation. We looked at moss, and we found slow millisecond and microsecond dynamics. This is slow for a protein. And we looked at green algae, where we saw microsecond dynamics. So one of the things that's interesting about thinking about these processes in different species is that we can compare them to figure out what kind of balance of these different processes gives us the best combination for things like increased biomass. And so from these studies, we were able to determine that some of the conformational switches in plants occurs in an associated protein. So these screens here are not really working. If somebody could fix that. They keep switching on and off. Thanks. All right. Well, we'll kind of wing it. Okay. Along with that, in moss, we can see that there are slower dynamics, which is consistent with the environmental conditions of moss. We know that moss grows in these safe and protected environments, damp and in the shade. And then finally, if we look at the proteins from green algae, we see much faster dynamics. And so today, as an example of our work, I'm going to tell you just about these proteins from green algae. also because we're interested in thinking about green algae for different biomass production. All right, so I showed you before this free energy landscape associated with the level of dissipation. And so one of the things that these initial experiments showed us is that we were able to identify a protein conformation with dissipation on and with dissipation off. Now, by doing some additional experiments, we were able to find that there's not just a single conformational switch in these proteins, but actually two different conformational switches, where there's two different structural changes to the protein that can turn the level of dissipation on. By a series of other experiments, we found that one of these switches is controlled by the pH drop, and the other of these switches is controlled by the carotenoid composition. This tells us that there's two distinct conformational switches in the protein, one controlled by pH and one controlled by carotenoid composition. By a lot more detailed analysis and experiments, what we're able to do is take this information and map it back onto the protein structure, where we're able to determine where in this protein the likely source of these conformational switches is. So from these spectroscopic experiments, we could put together this mechanistic picture of how these conformational changes occur. Along with this picture, we could do correlative measurements of what's happening in vivo. So we can measure the same parameter in green algae, where we're measuring again what's called the fluorescence lifetime, or for how long energy comes back out. The fluorescence lifetime gets shorter. That tells us that the level of dissipation is increasing. And what we saw in wild type is that dissipation increased under sunny conditions, as you'd expect. And then we looked at a series of mutants. First, we looked at a mutant that only had the pH-dependent dissipation, and we saw that we could recover the effect of dissipation. Then we looked at a mutant that had only the carotenoid dissipation, and we saw that regardless of the light condition, which changes the pH, the level of dissipation was maintained. So this is consistent with the in vitro experiments that show us that there are these two distinct processes to turn on dissipation in vivo. One controlled by pH and one controlled by carotenoids. And so we can conceive of this as the organism having two separate knobs, one where the level of dissipation is regulated through pH, and one where the level of dissipation is regulated through carotenoids. So thinking about this, we now see these two separate knobs responsible for tuning this control loop that I introduced at the beginning that sets the level of dissipation. So that's the picture that our spectroscopic and biochemical experiments were able to determine of dissipation and how it works in the protein. But now you may be wondering, why two? What is the evolutionary advantage to having these two separate control knobs for dissipation? And so to think about that a little bit more in detail, we can think more deeply about this control loop. So fundamentally, this is a regulatory process. If we think about a control loop, like what controls the temperature in this room, it's known as a PID loop. That means there are three separate components: a proportional, an integral, and a differential component. It's how temperature is regulated in rooms and how basically any control system works. And so the proportional component is proportional to the input. The integral component is proportional to the integral of the input. And the differential component is proportional to the differential of that input. Now, what we know from basic control theory is that if you have a fluctuating input, you need the integral control. And if we look at our input here, sunlight on a typical day, what we see is that it fluctuates. I mean, we also know this. We've seen clouds, right? And so that tells us that if we think about how we're going to regulate the level of energy in response to sunlight, we need integral control. So I told you that one of the dissipative conformational switches is controlled by pH. So fundamentally, what is pH? It's the accumulation of protons. So that accumulation of protons gives you a single integral control. So we know from control theory that a single integral control regulates a step change, like we see when a cloud or a shadow passes over something. Now, if we think about the other component, the carotenoid composition, we know from the biochemistry that the carotenoid composition is regulated by an enzyme. So it depends on the accumulation of that enzyme. But the activity of that enzyme is pH driven. So it depends on the accumulation of pH. So we're single integral control of the enzyme, but double integral control of proteins. And if we again turn back to control theory, a kind of basic loop like we have occurring in this room, this double integral control regulates a ramp change, something like sunrise or the breaking up of cloud cover. So fundamentally, what these studies have told us is that these proteins have evolved to have two separate conformational switches that activate dissipation. And by thinking about this in the context of the environmental responses, what we've been able to see is that these two conformational switches are able to respond to the two types of fluctuations in sunlight. All right. But I motivated all this by thinking about, can we actually increase the amount of biomass? Is there a way to increase the amount of energy? And so if we want to think about how to do that, we need to think about how things change when we turn these knobs or when the level of dissipation changes. So it's been well established for decades that you can knock out the enzyme responsible for the carotenoid conversion. So that knob, we have established strategies to control. But pH, which is part of the cellular feedback loop generated through photosynthesis, has been much more challenging to turn that associated knob. So to begin to investigate that question, one of the things that we did is went back to this protein and figured out a way to remove the section of it sensitive to pH. And what we were able to see is that removes the component associated with the conformational change or the conformational switch that activates dissipation. So, in thinking about this conformational switch that responds to types of fluctuations in sunlight, we've been able to identify the presence of these two separate switches that respond to the two types of fluctuations in sunlight. And whereas one of these switches has been well established, that you can control it in the organism, we've begun to show a pathway for a way that you can turn the other of these switches off. So collectively, the ability to turn these two control knobs provides us a platform to test hypotheses of how we would be able to set the switch to the ideal levels for increased biomass. Because if we want to think about ways to adjust the level of the slices of this pie that I introduced in the beginning, so this large component that leads to things like heat, can be reallocated as biomass. And so I told you just one story today of one aspect of the mechanisms behind the dissipation of excess energy as heat. This is just one example of how our fundamental knowledge of the complex biochemical and biological reactions of photosynthesis can provide us a pathway to develop a blueprint for the systems and structures associated with photosynthesis that may lead to this reallocation of energy into biomass. All right, and I want to spend my last couple minutes just making sure to acknowledge all of the people in the group that did this work. So there's been a lot of discussion so far about the importance of transdisciplinary work, and I want to start by saying that photosynthesis is inherently a transdisciplinary field where everything from quantum mechanics to agricultural science come together to understand how this works. A lot of the work that I told you about was in collaboration with the plant biologist Roberto Bassi, who's in the audience today, who provides not just protein, but really important biological insight and perspective. Along with Roberto, we've been lucky enough to work with Matteo Ballatari and Alberto Pinola as collaborators and colleagues on this work. And finally, much of what I presented today was developed by a former postdoc in the group, Toru Kondo, who's now an associate professor at Tokyo Tech doing beautiful work in his own group. And of course, I want to thank the funders, and in particular, the Human Frontiers Science Program that not only hosted this, but helped supported some of our more radical ideas about understanding this process. Thank you.
So thank you, Gabriella. We're going to continue the heat theme with Peter Franks from the University of Sydney. I was reminded in listening to Gabriella's talk and about the impact of heat of a comment from Joseph Conrad in The Heart of Darkness, the hazardous endeavour of living. And I think we're going to continue that theme now.
Thank you very much, Peter, and it's a real pleasure and an honor to be here. Thanks for the invitation. So, today I'm going to talk about plant heat stress, heat waves, and food security, and how those events are linked together. So these, what you see here, are some very familiar events that are resulting from climate forcing, from this climate change that we're undergoing at this present time. Damage to crops is an example of corn, which is particularly vulnerable to heat stress, heat waves, and declining agricultural output productivity. How are these events linked? And if we can understand those linkages, can we make changes in different parts of that system to affect other parts of that system at different times and in different places? So, just by way of a little bit of background, plants are very much connected to climate, and indeed plants and climate are very much connected to each other, and this is one way to sort of visualize how profound that connection is. If we look at a landscape through a thermal image camera, you can see the canopies of those oak trees in this particular landscape that have their roots deeply embedded in the rock fissures of the landscape, attached to water. They're transpiring, they're cooling the canopy, and that canopy is many degrees, several degrees cooler than the ambient temperature. The ambient temperature on that day was about 40 degrees, and you can see down in the riparian system down on the bottom where there's lots of water available, the canopy is much cooler than that, and the ground temperature, that bright orange, is much warmer than that. And so basically that principle, that applies across the global landscape. Different kinds of vegetation affect the climate in that way, with that mechanism by different amounts. So savanna systems or boreal forests, temperate forests, rainforests, their interaction with the climate and the climate affecting the behavior of those canopies and that heat balance and the evaporation from those canopies, they both affect each other, and they both change in different ways spatially and temporally across the globe. So, as has been mentioned previously, one of the advances that has been made in understanding the Earth system as a whole is including in part of that system a better understanding of the way the vegetated landscape interacts with climate. And that's basically what I'm going to be focusing on today. The real crux of that problem, or that question of how that process works, really happens down at the cellular level on the surface of leaves through these very important structures called stomata or stomatal pores. And through that process, millions of tons of water is evaporated into the atmosphere annually, and with that, the energy that's used to drive that process partitions solar energy into different aspects that affect climate differently. And so, to understand that process at the global scale, we really need to understand also what's going on down at that cellular scale, because all of those cellular components, those stomata scaled up 1 billion times, is what's feeding back onto global climate. And just to emphasize just how profound and important that process is, these structures, these stomata, they evolved about 400 million years ago. We had a bit of a time series history of the planet Earth yesterday, but just to look at the period of history, the last 800 million years in which plant life has occupied the continents, the first 400 million years ago was pretty uneventful, and it was mostly plants that didn't have stomates. And basically the surface of the Earth went through these wet and dry periods because those plants were very poor at managing their water resources. But as soon as stomates appeared 400 million years ago, and they gained this control over their water resource, and they became very efficient at maximizing carbon gain for the amount of water that they used. And they were so good at that that kind of drove the evolution of and appearance of global forests post-Devonian and into the Carboniferous. And in fact, it was mentioned yesterday that they were a little too good at this because they sucked out so much carbon from the atmosphere that they cooled the climate, and that had some downsides to it as well. So the question we're asking, back to this problem of heat and global warming and how that's going to affect processes and food security in the future. What happens when plants, when these canopies, whether they be natural systems or agricultural systems, when the canopy of those systems begins to fail and become stressed, because they're biological systems, so like all biological systems, they're vulnerable to breakdown under stress. And so we have in this basic concept of plants interacting with climate and these feedbacks going on. Plants affect climate, climate affects plants, and this has gone on for quite some time while we had a stable CO2, but now with that CO2 forcing, that's creating these two disturbances to that system. The CO2 is forcing climate, of course, but there's also a direct effect of CO2 on plants. Plants use CO2 for photosynthesis, and the plants are responding directly to that increased CO2 as well. So we have these two prongs of disturbance that are upsetting this feedback process that we have between plants and climate. So we need to understand more about how that process works so we can be better prepared to make predictions about heat waves and heat damage to crops, or perhaps even modify crops in the future to cope better with that. So the research we've been doing lately, and from funding from HFSP, sorry, Human Frontier Science Program, is how, well, it begins with, first of all, how stomates respond to CO2 and temperature. And so we know basically from observation, fundamental research begins with observation, and we know that if the CO2 is elevated, stomates tend to close to be more efficient at using water. They don't have to be open as far to get as much carbon if the CO2 is higher. And at elevated temperature, when temperature increases, they tend to open because it's advantageous to cool themselves. And so we wanted to know more about that process. And the first question we asked is, in fact, the first question I asked my molecular colleague in this process, I said, well, what genes do we have for understanding heat response of stomates? And he said, well, we don't really have any signaling pathways. We know about blue light, we know about CO2, but we really don't know anything about the heat sensing and response of stomates. And I found that rather remarkable. And that's an example of a classic example of a knowledge gap that needs fundamental research. And so we've begun to embark on efforts to uncover that pathway of heat response, and using some techniques called loss of function in plant models in Arabidopsis, we've begun to make headway into uncovering parts of that signaling pathway. And what we've found, I don't have time to go into the detail, but in principle what we've found, we've uncovered a new kinase that is part of that pathway, but what we've discovered is that receptor kinase actually is common to both the signaling pathway for CO2 sensing and temperature sensing. And so there's a little bit of a nexus there, and the problem is it creates this dilemma when both of those environmental signals are going in the same direction. When both are increasing, the plant is faced with this dilemma of whether it closes down to be more water use efficient or whether it opens its stomates to be a little cooler under excessive temperature. And that's probably not too much of a dilemma while temperatures are more moderate, but as soon as temperatures become more extreme, that then puts the plant into a different, a more challenging problem to solve, and more of a dilemma. And so stomates generally, much of our understanding of stomatal function is based on this principle of optimization of carbon gain. So there are lots of models that explain how stomates optimize that trade-off between gaining carbon and losing water. But we haven't really solved the problem of when you put heat over the top of that problem, excessive heat, how the plants cope with that. So that's an ongoing part of our research. The second part of our question is, how can we manipulate, if we know something about that control system, can we manipulate that system in a meaningful way that will give us information about global climate change? Can we implement that knowledge somehow in global climate models? And so global climate models, as you know, they run on these massive supercomputers, and buried within the thousands of lines of code, of course, are models. And all of the talks that you've heard today are basically models. All our understanding of the world around us is basically models, but we need good models and reliable models. And one of the questions that we're asking is, this model, so GW there on the left is basically the stomatal conductance. It's basically a representation of how wide open or closed those stomatal pores are. And this is a simple model. And the other thing is models need to be practical. So there's a trade-off between a really complex and accurate model and a model that's going to be malleable and amenable to usage in something as large and complex as a global model. So we are constantly searching for the optimum useful model. But just assuming this model, this model has its own, it had, like all models, it has simplifications, assumptions, but let's assume that it's an okay model for now. Most of the parameters in that model, all of them except one, are either climatic variables or biological variables. But there's one in particular that, and this is a common feature of many models, is that there's a parameterization factor. And the way that we currently parameterize that model to account for the differences, those differences in different type of vegetation around the globe is we change the value of that G1 constant. And so traditionally that constant has been prescribed and based on very limited physiological data. So there are ways to go out with instruments and actually measure on a single leaf what the G1 value is for a particular type of vegetation. But the problem with that prescription approach is that that's very static. And when you've got a changing climate and a changing vegetation, with a static parameterization, how is that going to cope with the change with simulating a changing climate. And so that's the other idea I want to introduce here is the models need to be realistic representations of a system. It's not enough for them just to work and give them the answer that you want. Now back to that parameterization feature of that model. What we've been able to do, well, it's been actually known that it's a somewhat enigmatic or mysterious parameter, but it's basically linked to the water use efficiency of plants. And if you were to map, so the water use efficiency is, in simple terms, is the amount of carbon that's fixed for a given amount of water. Or if you want to use rainfall as a proxy for how much water is available, and you plot productivity, global productivity versus rainfall, and what we see is across the globe, there's this non-linear response, non-linear relationship between productivity and rainfall. And basically that means that plants have different water use efficiencies in different world biomes. And so what you might expect if you were to visualize that, of course, in tropical rainforests where water is abundant, the water use efficiency of those plants is quite low, and then plants get more and more efficient at using water the drier the environment gets, down to the most arid there in the left-hand corner. And so what we've been able to do is we've been able to develop theory and show empirically that there is a pretty good solid relationship between this G1 parameter and rainfall. And so when we use rainfall to parameterize, instead of doing it statically and somewhat subjectively, as we have in the past, if we take gridded rainfall and translate that to this sort of a continuous G1 value across the landscape and run it in global simulations, it actually, it not only works really well, so we've validated this with data, and it actually, the simulations work just as well and probably better with the use of this technique. So the example on the far right, so the left-hand example, the left-hand example there is the default, and then we've got two different versions of increasing complexity of applying this mean annual precipitation technique. And you can see by this overall, this standard Euclidean distance is a metric that you can use to basically give you an overview of all the uncertainty and error in the model, in the simulation models. And you can see that the lower that value, the better the simulation. So we have something more realistic. We have a parameterization process that is linked directly now to rainfall, so that when climate changes, that parameterization can move with that changing climate. And so that it's more in tune with our understanding of the way that climate and vegetation not only interact, but how they co-evolve together, whether it be over periods of millions of years or the next several decades. But the real value in this is we want to be better at projecting. We want to be better at understanding what the climate's going to be like, say, for example, at the end of this century or the next century, sometime into the future. And so we need reliable models, and we've done some simulations, some projections with this technique, and just by way of example, ask the question, well, how does that affect, for example, projections of heat waves? Heat waves are a very real threat in future climates. It was said 20 years ago by David King that record-breaking heat waves are going to be the norm. But we need to know when and where those heat waves are going to occur globally. So we need to get better at spatially and temporally predicting those. It's okay. I think we're all resigned to the fact that the climate's going to be hotter, but where exactly is it going to be hotter? And so with this new technique, I think we have to revise some of those projections. And for example, when we run, this slide shows when using that new rainfall-based method, it shows that there are some regions where there's improvement, where there's less heat wave days. That's the areas in blue, and there's some areas where heat waves are going to be more intense or longer. In this case, they're going to be several weeks longer, several weeks more days of the year where there will be heat waves. And we're talking about, with heat waves, these are lethal events. We're talking about the difference between saving lives or losing lives. So this, even changing this information by a matter of weeks will help in that pursuit, but also help direct resources, whether it be civil infrastructure, medical facilities, agricultural investments in agricultural techniques, biotechnology to genetically alter crops. Where should those efforts be focused more tightly? So basically, the conclusions from this, I'm just going to make some very overall general conclusions, and the first couple, of course, are obvious. Plant heat stress is a critical threat to food security, and plant regulation of water, carbon, and energy fluxes, we need to be better at predicting those processes. And large mechanistic gaps in this understanding can be filled using fundamental science. And so I gave 2 examples. So that the genetic pathway for controlling that stomatal regulation, that can be used as that is an example of the fundamental science. But also our models need to be conceptually realistic. So we need to move away from sort of mistakes and habits that old habits that we're comfortable with, perhaps with older models, and think about new ways of using perhaps even those same models or developing new ones. And so basically this only will come through scalable and integrated approaches with transdisciplinary research and being encouraged by organizations like this to reach out and work across different fields with different people, perhaps ideally people that haven't worked together before. And so a big thanks to the Human Frontiers Science Program and to my colleagues, Sydney, San Diego, and Exeter, Stanford, Purdue, and NCAR, who are involved in just this specific project. Thank you very much.
Thank you for that, Peter. I think that last line, that what we need are integrated, scalable approaches, and those aren't easy to find, is something that's going to be a theme through a lot of this meeting. And I think it's also a good link into our next speaker. So I'm pleased to introduce Amy Rowett from UCLA, who's going to talk to us about some of those scalable approaches to delivering different types of meat production.
Thank you very much. All right, well, it's a great pleasure and honor to be here today to tell you about some of our latest work in developing novel approaches to producing animal protein in a more sustainable way that has sustainable potential. All right, so the vision that really drives our work is to address some of these major societal challenges, which we've heard much about in the last couple of days. This very intertwined challenges of advancing human and planetary health along with social justice. And in my own work, I'm doing that. So the major research question that drives our work is to really understand how cells respond to physical mechanical cues in their environment to generate an integrated response. And if we could fully understand the molecules and pathways that are involved in regulating cellular response to these mechanical cues, it would really enable us to control and also predict cell behaviors for a spectrum of applications spanning human and planetary health. And in my own lab, we're addressing this question by building new technologies and platforms to be able to measure in higher throughput cell mechanical behaviors. We're working to understand the fundamental mechanisms that regulate these behaviors, and also translating these findings for applications in both cancer and also in sustainable food. And that's what I'm here to tell you about today. So the vision for, that really drives this arm of our work, which we've been working on for the past few years, so it's a newer direction in our research program, is really to develop nutrient-rich food that can be available and accessible for everyone. Meat is a very nutritious food for many people around the world, but in the face of extreme climate events or supply chain disruptions that many of us have experienced over the past few years, we're at risk for having nutrient-rich foods become very expensive and a luxury item. So having sort of multiple sort of ways of producing animal protein could help to build resilience in our food supply chains. There's also increasing demand from consumers these days for foods that are both nutritious but also support planetary health and have reduced environmental impact. And as you can see from many of you've experienced the emergence of plant-based meats, which are very popular, but the majority of consumers still want to eat real meat. So this is where cultured meat, the process of using cells extracted from a living animal and growing them in culture to form meat products, can be a potential solution. And there's still lots of work ongoing to examine the environmental implications and also technical feasibility. But our data so far is suggesting that cultured meat can be a potential solution to creating a sustainable source of animal protein. It's been mentioned also the IPCC report, which also sort of highlights the importance of novel foods to address some of these climate goals as well. Cultured meat may strike you as a new idea, but it's been around for quite some time, originally described by the concept by Churchill, which really kind of got at the question of why we couldn't grow just individual pieces of meat without the whole animal, which can be more energetically wasteful. But fast forward nearly 100 years, and you may have heard this news last week that in the United States, the USDA has now just approved the sale, making it the second country after Singapore to be selling cultured meat products. This may give you the impression that we've solved all the problems. We haven't. This meat is only available at a very small scale, and so there's still major challenges to be solved in research and development. So to give you a sense, typically we culture cells in a very small petri dish. The total mass of cells in a petri dish would be on the scale of milligrams, but to produce cells for food, we'd need orders of magnitude higher mass of cells. And this can be achieved by just using a larger number of petri dishes, which is what was done by Mark Pust, who pioneered this field back in 2013 for the first proof of concept cultured meat burger. Of course, this is not sustainable. So growing cells in larger bioreactor vessels is a very attractive solution. So there's various challenges in culturing meat on such a large scale. People in labs around the world are working on addressing the media, the source of cells, and also how do we generate foods that people would want to eat, that are delicious and that taste delicious and have a great texture. And with my background in mechanobiology, this is exactly the question that we're seeking to address. So we know from foundational work in mechanobiology that these mechanical cues that regulate cell behaviors are really important for driving some of the phenotypes important for cultured meat. You can see from this work from Penny Gilbert that cells, muscle cells taken from mouse grow much more extensively on a softer 12 kilopascal hydrogel rather than on the stiffer petri dish, which is shown on the bottom. We also know from work of Adam Engler and Dennis Disher that myoblasts cultured on sort of medium stiffness substrate of around 11 kilopascal are more likely to fuse and form myotubes, which are important for the development of skeletal muscle. As you can see from the alignment of these blue nuclei. So taking these findings, we've been developing customized scaffolds to grow both muscle and also fat tissue for cultured meat applications. So to give you an overview, our approach is to make tiny scaffolds that can be also compatible with culture in a suspension bioreactor. And the idea is to harvest these tissues into a meat product. So we use emulsions, just the way you would make a salad dressing, water and oil, to form a template for these tiny microbead particles that look like this. We can control the stiffness just by the amount of polymer that's in these beads. For proof of concept, we've used gelatin. We can also replicate this grooved structure like natural skeletal muscle by making this template that we sort of squish the beads between. This makes these grooved microcarriers, which we can also modulate the stiffness of as well. So the question is, can the cells grow on these? And we use mouse CTC12 cells as proof of concept here. You can see that the cells, the gelatin is shown in yellow. The cells have cyan-colored nuclei, and we compare these to commercially available cytodex beads. Over time, we see that the number of cells tends to increase over time, suggesting that cells are growing very nicely on these edible microcarriers. And we've quantified the growth of cells by quantifying genomic DNA as well, which you can see that the cells on the edible microcarriers shown in pink and blue are increasing in number even more compared to the commercially available microcarriers. So can these cells also differentiate to form the beautiful myotubes that are important for muscle structure? And shown here in the magenta is a myosin heavy chain stain. So you can see these myotubes actually can span hundreds of microns through these aggregates of cells and these microcarrier particles. It's not quite like the skeletal muscle shown here by the rat, but they're much longer than the cytodex commercially available microcarriers. What was interesting is that we found these aggregates of these microcarriers and the cells forming in suspension culture, and you can see across the different edible microcarriers, these structures forming. Again, this is with mouse cells, so who really wants to eat mouse cells? There is a market, no joke, for pet food, cultured pet food. But we also tested the use of bovine satellite muscle cells and showed similar effects. We're able to harvest these microtissues into a larger cohesive structure that can be cooked, shows signs of browning characteristic of myoid reactions. So what about the fat? We know fat is really important for the flavor, the mouthfeel. This is just showing the sort of characterization of Wagyu beef with increasing marbling being more desired. You can find this in cultures around the world. So we also were able to culture cells, both primary rabbit adipocytes and mouse 3T3L1 adipocytes on these beads. The beads are shown in cyan and the nuclei in blue. And the lipids you can see accumulating in yellow for both the rabbit and the mouse. We were then able to integrate these fat microtissues into layered structures with our muscle microtissues. And you can see here a cross-section of cultured rabbit meat where the blue is showing you the protein component, the muscle component, and the cyan are those microcarriers from the fat component. Shown on the right is false color, just for easier visualization, the fat in yellow and the red is the muscle. Compared to Wagyu beef steak, where you can see a similar layered structure, albeit the length scale of the muscle component is different. But importantly, this is proof of concept work for our cultured meat that shows the feasibility of creating these these structures. And what's really interesting is that we found these multi-component structures happen by spontaneous adhesion between these microtissues, which results in a cohesive, mechanically stable structure. And we've begun to mechanically characterize these cultured meats, comparing them to Wagyu beefsteak, and we are seeing similar on the order of magnitude mechanical properties, for example, ultimate tensile strength. The scale is still small. You can see by the size of the sesame seeds on the bun, but we're working to scale up, and that's really, I think, the realm of more commercial opportunities. So today, I've shown you that we can grow muscle and fat microtissues in suspension culture using these edible microcarriers. I think this has potential as a scalable process for culturing cells for food applications. We're able to generate these multi-component tissues and pattern these tissues just by spontaneous adhesion. And I've shown you we can translate the approach to different types of cells as well. Beef, rabbit, and mouse. I didn't show you some of the work from our pectin microcarriers, but we see similar behaviors on plant-based microcarriers as well. So I think another exciting opportunity here is to highlight some of the more fundamental research questions that come out of some of this work. For example, how do these principles of mechanobiology translate across species? A lot of the optimal stiffness that's been discovered for accelerating cell proliferation and differentiation, for example, it's been largely conducted on mouse models. And so I think here there's opportunity to understand how broadly do these principles of mechanobiology apply across different species. Furthermore, I think it's really will be essential into the future to understand how cells integrate different signals from different mechanical stimuli, for example, the matrix stiffness, together with the fluid shear stresses that cells might experience in suspension culture. So this is towards our vision to be able to create cultured meat, both ground meat and mixed, but also structured cuts of meat. And I'd just like to highlight some of our ongoing work that I think really uses and translates some approaches from the life sciences to address these questions. Specifically, we've been working in long-time collaboration with Robert Damoiseau of the Molecular Shared Screening Resource at UCLA. So we've been now screening libraries of small molecules and potential food ingredients to better understand how we could what sort of soluble additives we could include in the media, and also to understand more fundamentally the mechanisms and pathways that might be involved in regulating, for example, lipid accumulation in adipose tissues. Of course, there are many outstanding challenges remaining. This one's come up already, is public perception and consumer adoption. And so to address this question, we're working with our behavioral scientist collaborator, Janet Tomoyama at UCLA. And what's interesting, I think, to us is that there's lots of challenges one can envision for these novel food products, but also challenges that we can address through fundamental research in the life sciences and also further research and development, such as understanding how we can make a better tasting or a better textured piece of cultured meat. Last but not least, I think engaging people in critical conversations about novel foods to encourage this transparent dialogue will be really critical. And to this end, I've been hosting these events over the past 10, 12 years now to engage public in these conversations. Here's one on people, food, and climate that we hosted last year that brought together architect, a chef, and heirloom corn entrepreneur and regenerative agriculture scientist and soil scientist. So you can find these available on YouTube, Science and Food on YouTube. So just to quickly finish, acknowledging some of the people, Stephanie Kveski here, graduate student, has really been a major proponent and spearheading this direction of research in my group. And of course, funding sources specifically for the cultured meat, the USDA, the NSF, and also the philanthropic sources such as the Nobel Family Foundation Fund at UCLA. Thank you very much.
That was fascinating. I can't get away from the concept of a mouse steak, though. But we've heard now some really quite spectacular developments and technologies that have the potential to have an enormous impact. The question that then arises, do we have enough? Do we have technology? Do we have capabilities and systems in place to actually achieve food sustainability? And that's a question that Jean-François Susanna is going to address now. Jean-François is Vice President for International Relations with INRE here in France. Jean-François.
Thank you, Peter. Yes, thank you, Peter. So I will try to take a step back and explore how we could get food system transformation towards sustainability. Well, we have big challenges, and the first one is how could we end hunger? This is the target of the Sustainable Development Goal 2, SDG 2. However, as explained during my pitch on the first day of this conference, we have to understand how this could also connect with other challenges like health, like water, like biodiversity, climate. So it's really a nexus of problems we have to solve by changing the global food system. And this relates, in fact, to the 17 SDGs. So it's really a major undertaking. This was the vision of this Food System Summit organized by the United Nations in 2021. We will have a stocktake of the summit in August. And there are 5 specific objectives here. Ensuring access to safe and nutritious food for all, shifting to sustainable consumption patterns, boosting nature-positive production at sufficient scale, advancing the equitable livelihoods and their value distribution, and building resilience to vulnerabilities, shocks, and stress. So you see how, let's say, the global community framed those 5 challenges. It's quite interesting to see. Let me now explore with you what is this landscape of problems we are having with the global food systems. I will just take a few examples. The first one is really that we are losing a lot of food. We have to realize that the food system includes the production, processing, distribution, consumption of food, and the output of these activities with their implications for socioeconomic, environmental dimensions at the global scale. So we are losing a lot of food, and we are also with very large problems with malnutrition. Malnutrition is often called a triple burden because it includes undernutrition. So we have around 820 million people worldwide that are chronically undernourished. Micronutrient deficiencies, that's about 1.2 billion people, like not having enough zinc, enough iron, or enough vitamins. And we have an epidemic of overweight and obesity, which is actually leading to a very large burden in terms of chronic diseases. So right now, this is a leading cause, malnutrition, of illness, disability, and mortality. 1 in 3 suffers, and it will be higher in 2030. It's estimated 1 in 2. So we have a huge problem there. I address this issue of food losses and waste, but it also contributes to around 10% of the anthropogenic greenhouse gas emissions. So if we were to reduce this, and let me say that the losses are mainly in the low-income countries where you have problems with the logistics, with the conservation of the commodities, and the waste they are mostly in the high-income countries where we tend to overconsume and waste quite a lot of our food. So we could certainly address this, and this would have huge benefits for the environment also. Then we need to consider this malnutrition issue. So it's not only tackling the food access, which is a key issue for undernourishment. It doesn't, in fact, require a lot more food to be produced. It's really an access problem in our days. But it is also rebalancing our diets with more fruits, vegetables, so more fibers for your microbiota, more protein crops and nuts. And this would come with actually rebalancing the production systems, going towards integrated systems, diversified crop rotations, increased genetic diversity, and the resilient and low-emission livestock systems. This would certainly support climate change adaptation and mitigation. So if we have this perspective where in fact we need to transform our food system to have healthy and sustainable diets, we realize we can do a lot. Because, for instance, if we reduce in the high-income countries the consumption of meat, the consumption of milk, because it is in fact a bit excessive compared to the nutritional recommendations, we could certainly have more land for ecosystems or for bioenergy, and this would in fact release millions of kilometers square of land with co-benefits for environment and health. So let me now introduce the vast risk we have with climate change. This is really a risk to food supply stability. This is a well-known graph from IPCC, which is the burning ember graph. So you see those colors going to the purple when you have increased warming. So far, we are very close to, unfortunately, the 1.5 degrees Celsius target for warming. We're already at 1.2 degrees Celsius. We are really at risk of exceeding 1.5. And already we see infrequent price spikes that affect individual countries. So indeed, climate change with droughts, with heat waves, is reducing the access to food and creating price spikes already. If we move towards 2 degrees Celsius and more, then we will have regional shocks because there are multiple bread baskets that are in fact interconnected through climate and may be affected, and this may really rise the price for entire regions. And if we move beyond that to 3 or 4 degrees Celsius, we are really at risk of having our global food supply disturbed. So you understand this is a major risk. I referred to also the micronutrient deficiencies. Well, that's a bad news because increased atmospheric CO2 actually leads to further reductions in the micronutrients in grains, and this means this will aggravate the nutrient deficiencies. We have some problems with economic models to capture all this because they often rely on general equilibrium as an assumption, which is not what we observe. They capture a rather modest increase in food prices, but what we observe is that, in fact, when we have heat and drought, we have conflicts, we have migrations, and this aggravates the food security issue. And we know certainly that the most vulnerable people are, in fact, more affected. So what can we do? First of all, we need to fight climate change for sure, and the global emissions from our food systems are around 30% of the total anthropogenic greenhouse gas emissions. So we need to deploy response options across the entire food system from production to consumption, including, as I said, the food losses and wastes, and supporting adaptation and mitigation. So we will explore a bit more the landscape of response options we can see, and it is a large mitigation potential there between 2 and 9 or 10 gigaton of CO2 per year. The uncertainties are high. There are more with, in fact, how much adoption we can get with those technologies. But before exploring these responses, let me say that the challenges with the land systems are huge because we have challenges in terms of climate change adaptation. In those graphs at the left, you will see some, is this a pointer? Well, sorry, wrong button, okay. Yeah, let me say that on the top graph at the left, in purple, in highlighted colors, you have the risks linked with climate change adaptation, so the impacts of climate change. And the risks are, in those areas, higher because the seasonal cycle is disturbed by climate change. Then you have desertification, land degradation risks. You can see they are highlighted on the maps, mostly in tropical areas. Then you have also the food security issue, where we have countries with chronic undernourishment, the biodiversity hotspots, which are threatened, and then at the bottom you can see those areas where we have water over-abstraction and also where we have excessive nutrient flows by nitrogen and phosphorus. So if you want to add those challenges that are overlapping, you get to this map where you see that there may be 4 or 5 challenges, especially in the tropical countries, at the same time in the same region. it is interesting to note that the smaller the Human Development Index, the larger the number of land-based challenges per country. So it is really in countries that are already experiencing problems with health, problems with education, problems with GDP, that you have the largest number of challenges. So this also shows the conundrum and the problems we have with that nexus. So we've been exploring this IPCC report, special report of IPCC on climate change and land, the response options in a systematic way, so I cannot explain all of this, but you see they are organized in terms of agriculture, forests, other land uses or other ecosystems, and then in terms of the response options based on the value chain management and response options based on risk management. Without entering into the details, the message here is that whenever you see a blue color, there are multiple benefits. So, in fact, if you take a single response, like the increase in soil organic carbon, that response is always colored in blue. It has co-benefits for mitigation, adaptations, for desertification, land degradation, and food security. So we should, as an example, deploy that response on a large scale and urgently. But there are many other examples here. So we can do a lot, in fact. There are a few cases where you have negative implications, and this is often with food security, where, in fact, if you use more land, you may compete with the use for food, and that's a sort of trade-off between food and environment. So here again, without entering into all the details, you see how we can plot, we can map the responses in terms of their potential deployment. These are the land management responses, and when you see a color, this is a region where apparently there would be only co-benefits or no harm in deploying that response. And when the color is black, is that it is not adequate to deploy that response because we see adverse effects. So here again, in many regions, we could deploy a lot of responses with mostly co-benefits. However, you may see that the last map at the bottom and right, it is about bioenergy and what is called BECCS, as it is the bioenergy with capture of carbon and sequestration. There are few world areas where we could deploy this without harm, and the reason is that deploying this implies trade-offs with biodiversity and also with food security. So this is the graph I used during my pitch on the first day to see that, in fact, when we combine the responses we have in terms of agriculture, bioenergy, nature-based solutions, food access, and food sufficiency, we may still be able to reach the SDGs by 2050. But please consider we need to do a lot, and in only 25, 30 years, and it implies working in a coordinated way between the agriculture, between the environment sector, the climate policies, and also the food policies. So policy coordination internationally, but also within countries, is certainly something that needs to be highlighted. So now I'll move to discuss a bit more what are the innovation pathways and also what are the policies we can see in some countries. This is a graph we have at INRAE to explain how we want to shift towards carbon neutral and climate resilient food systems. You see that climate resilient, we can do quite a lot by having some breeding technologies used. This can be for plants, can be for animals. It is about breeding, phenotyping. Then we need to have soil and water conservation and to diversify the farms to mitigate the risks and have new value chains. For the carbon neutrality, it's obviously about abating the greenhouse gas emissions. It is also about soils and biomass carbon. It is also about bio-based inputs and supply chain efficiencies. So if we combine this, plus reduce the waste and losses, plus rebalance the diets, we get pretty close to carbon neutral and climate resilient food systems. And indeed, we are starting to see policies, for instance, in France about this, about transition towards agroecology, which is sort of driving targets that we have for the agricultural policy, about climate resilience with adaptation plans, with plans about water resources, and also about carbon neutrality We have indeed a carbon neutrality strategy in France by 2050, and each year we have some carbon budgets that are established and should actually be according to the national strategy. So we also have reports. I'm in the French National Climate Change Committee. We are an independent committee reporting on how we are achieving those targets. It is therefore important that in that transition we have strong interdisciplinary research programs. That's an example of a research program we have at INRAE about climate change. And we said that it would be most important to understand how to combine in our sector with agriculture, forest, food, adaptation and mitigation. And this is in fact understudied so far. You have a lot of literature about climate change adaptation, a lot of literature about climate change mitigation, but you will not find much crossing those two dimensions. So there are really scientific frontiers in understanding how we can combine those, and we need to prioritize also this systemic transformation approaches and move towards transdisciplinarity involving the stakeholders. To explain why we need to do this is just one example, and I did not plot the most recent year, which is 2022, but that's the example of 2017, where we had a 30% decline in wheat yield in France, 20% drop in cereal production. We had about the same last year during summer 2022, and it could be as in that slide, because in fact of excess rainfall in spring, with problems with local flooding and disease pressures from the fungi, but it could also be because of heat and drought in summer, as we experienced last summer, 2022, or in 2003. So we are really in a difficult position because we should adapt crops to both the heat and drought and to both the risk of heavy rainfall and flooding. So how are we going to do this? It's a bit unclear. So that's why we need to increase also farm resilience. Traditionally, the thinking has been, let's do it with precision agriculture and advanced breeding. And this is, in fact, keeping a bit the same system and improving it. We call it incremental adaptation. However, as I said, it's quite difficult with breeding only to understand how you can have the best plant material for heat and drought and for heavy rainfall. So we have to think a bit possibly about something else. And that's when we think about systemic adaptation. That's about soil and water conservation. In France, we say it's about agroecology also, meaning that we want to use better the natural systems regulation and use less inputs to rebalance the systems. So certainly by improving your soils, by having integrated water management at catchment scale, conservation agriculture, crop livestock integration, perennial crops like in agroforestry, you can move towards more resilient systems. And there is also a big challenge about transformative adaptation, which is really rethinking what you're producing and also combining it with the changes in the food consumption with the healthy diets. So there you see we could have some grass-legume mixtures, we could have more of the fruit trees with agroforestry, some nuts and so on. We could have diversified landscapes which would reduce the pest and disease pressure, and that's really connected to the food system transformation. However, it is not really supported strongly by the current policies. So that's just an example on how you're going to mix the crops with crop mixtures, with the grain legumes introduced, and this shows we have, in fact, many examples ongoing. With livestock, it's about breeding the robust and resilient animals and improving the welfare for the animals, closing the nutrient cycles, providing the livestock sector with more services, matching the societal expectations, and we can do quite a lot, for instance, by having adaptation of the breeds to heat, and this thermotolerance will be very helpful in the future. So these are examples of plant breeding for heat and drought in France that run from genomics to phenotyping. And we have advanced phenotyping facilities which are both indoors and outdoors, with possibility to combine with actually the rain shelters and high CO2. These are examples about using, oh sorry, that's moving too quick, using the plant and animal diversity. So certainly the way we can conserve the biological resources is essential. We have a large national infrastructure about biological resources for plants, animals, but also for microbes. And there are European projects that are testing innovations in plant varieties. So it is important we not only rely on the modern pools used for crops, but we also rely on a much broader pool of genetic resources to face climate change. That's another example on how we can develop more environmentally friendly vineyards. You know, that's important for us in France. So we have to consider how to use biocontrol, genetic resistances. We have introduced some genetic resistance in some of those vines, and now we use 80 to 90% less of the fungicides, so it's really impressive what you can do. You can also work with the plant management in terms of architecture, of microclimate, and that's also converging towards climate change adaptation. Another example on what you could do with the plant-microbe interactions. So root phenotyping is something important for the future, and my slides are moving too fast. And here is, I will just move back. You see, we can address this, and actually you can have progress with this on the development of nitrogen fixation, and this is quite important for developing more grain legumes and more pasture legumes to reduce the inorganic nitrogen fertilizer inputs. So that's one aspect, but if we go back to, let's say, how we can, in a low-carbon agriculture, manage the carbon pools and the greenhouse gas fluxes at the farm level, that's a slide which is possibly well known to some of you, showing the diverse sources of greenhouse gases in a farm. So I said you could reduce with legumes the nitrous oxide. You could certainly reduce the losses of fertilizers like ammonia volatilization. But when we did a study, this is a national scale study for French agriculture by 2030, we see there are a number of options we could deploy. And it's interesting, on this graph, you see the options like saving energy, increasing grazing, using and fixing legumes, or reducing the fertilizers. Well, it's interesting to note that the first part of this graph, according to expert calculations, comes at a negative cost. So mitigating by saving inputs and reducing production costs is clearly an option. Then you have other options that you can deploy that have a moderate cost. This could be also with reduced tillage, conservation agriculture. We are subsidizing biogas, so it is with a relatively moderate cost. And some options come with a higher cost. This is about cover crops, agroforestry, and so on. The potential is quite high. With those options, we were estimating we could abate about 30% of the global greenhouse gas emissions without changing the way we produce, without changing the mix of crops and livestock. I would like to focus now on storing organic carbon in soils because this has a huge potential. How can we do this? We can do it by greening the soils, if one can say. So having a continuous cover by vegetation, introducing cover crops, introducing grass leaves, for example, is very effective. And in this way, you can also protect soils against erosion and degradation. This requires, however, some assessment. We could assess that we can store 30 million tons of CO2 equivalent per year, which is really high for France, especially through those three practices: cover crops, grass leaves, increased organic fertilization. And we see that the potential is higher when you start from a depleted soil. That's where you can regain most carbon. And the costs are actually compatible with what can be done now. Around median cost was around 55 tons per ton of CO2, 55 euros per ton of CO2 in 2020. The big challenge we have now, and it is undertaken also by the European Commission, is to create a methodology for monitoring, reporting, and verifying the soil organic carbon change. We need to deploy that methodology if we want the private sector to pay carbon credits to farmers based on results. So that's a paper we had where we could show how to combine, in fact, the different data sets, the long-term experiments at benchmark sites, the flux data we have with flux towers, eddy covariance, with shorter experiments, the remote sensing, which is very important to verify the data, the activity data, and then the modeling, which is driven by inputs about climate, soils, land cover, but also by the farm activity data. So when you combine this, you can readily calculate the soil organic carbon change over time, and this sort of combination is really discussed to see what would be the framework in Europe and internationally to have a standardized MRV. It is interesting to note the progress we have with remote sensing, with the Sentinel satellites that have a 10-meter spatial resolution and a weekly update. We can actually show what are the crops, where are they, what is the vegetation cover, therefore what is the amount of carbon returning to the soil, and from this, with the farm data, we can calculate from the field to the country scale the soil organic carbon stock change. So this is really going to be, I think, deployed on a large scale. We also have some, let's say, integrative projects. This is one Horizon 2020 project, which is looking into how could you get carbon neutral farms. Well, you could get there with a number of actually practices combined, but certainly with efforts like living labs, where you accelerate innovation by having an innovation which is co-designed between the stakeholders, the farmers, the value chain, and the researchers. So this is the intention here. It is gradually deploying over a number of countries in Europe and over a number of contrasted farming systems and value chains. So let me stop here with my take-home messages. We do have a solution space for land and food transition. It is narrow and it requires coordinated research and innovation across 3 dimensions. Having resilient and low-carbon agriculture within a circular bioeconomy, having nature-based solutions and agroecology, which is a form of nature-based solution that certainly include soil health, forests, wetlands, peatlands, and having improved food access in the low-income countries, and healthy diets and food sufficiency. Food sufficiency meaning you don't need to consume too much, you just need what is needed for your health, and this leads to also rebalancing the diets. So there are many technical options that are available, but they are often not used because of past dependencies. Transitions need to be better coordinated, and that's one of the main reasons they are not deployed. It is because if you want to have transition both in agriculture, food, and the environment, it needs to be coordinated. This requires a way we rethink our priorities, and certainly health, climate change needs to be at the heart of those priorities, environment also, while supporting farmers and poor consumers. And this requires strong international commitments, strong policies, along with private sector engagement. Research and innovation need to be refocused with system thinking, inter- and transdisciplinarity. Living labs are a good example, support to public policies, independent assessments, while providing innovations to facilitate transitions. Thank you.
So thank you for that amazing big picture approach to the problem. We're going to follow it up by taking a look in a bit more detail about how theoretical biology can help, with a talk from Chaitanya Gokali from the Center for Computational and Theoretical Biology at the University of Würzburg and the Max Planck Institute for Theoretical Biology.
Thank you. Yeah. Right. Yes, so I mean, I would be thrilled if there is a Max Planck Institute for theoretical biology. Unfortunately, it's only for evolutionary biology, but I hope you can hear me well. Right, so my name is Chaitanya, and I'm trying to follow up like a very big concept from over there, but trying to condense it down to something for theoretical biology and agriculture. So let me first go ahead by just mentioning what I mean by theoretical biology, because it might not be very clear to everyone in this audience, because it somehow might seem quite far away, because we have heard a bit about models, right? So what we try to do in theoretical biology is definitely do come up with models, but one of the fundamental things that we want to understand, at least in my group, is to develop theories to understand these processes which are common across scales in which living systems are involved, right? And then try to, if possible, wherever it is possible, to develop some translational applications. And let me just mention what I mean by that. So by fundamental studies, we try to look at all the way from the abstract models, from the origins of life, all the way to societal developments, like how do societies behave and concepts work in social evolution. But then at some point, you can also translate this understanding into actual applications. For example, we have been working in Darwinian medicine, Darwinian agriculture, and then trying to push a bit more into conservation and policy making. And that only happens exactly as you mentioned, by working across fields and working together with the people from who have different expertise than in these different fields. While we are doing this, of course, we want to maintain a healthy toolbox, because as you go across these scales of organization, which are massive, for each of these different systems, one needs to have a different tool to use. And yeah, we try to focus on a few of these tools, but then we keep to use one or drop one, depending on what one needs, right, for the relevant question at hand. And the question that we would like to focus on over here, as we all are doing, is sustainable agriculture. And I know that I'm going to be preaching to the choir here. You know everything about the problems that we have in achieving sustainable agriculture, because what we want to achieve is pretty broad, right? We want to have profits for the farmers, because otherwise that's not societally correct. So we need it to be societally sustainable as well. But at the same point, it also needs to be ecologically and evolutionarily sustainable. And I'll come to what I mean by that actually in a bit. And this is exactly what I mean. Like you know this far better than I do, that we are using like half of the habitable land just for agriculture. And the impact that it has actually on the environment is massive. It goes all the way across greenhouse gases, as we heard, to the biodiversity of animals that we see, which is mostly driven by the livestock that we have. So the impact is undeniable, but then how do we work towards yet doubling the output that we need? Because yes, the population growth is slowing down actually of the world, but it's still estimated that it will reach its peak by 2080. And this is a much more immediate problem then, right? If we want to still double the yields, and we already saw that the burden is way too high. So how do we actually manage to do that? Well, it is a complex problem. You have economics involved, you have ecology, you have evolution. I'll mention why, evolution as well. But if it's such a complex problem, how do we deal with it, right? Like, I come from doing mostly theory. Theoretical biology takes a lot of inspiration, let's say, from theoretical physics as well, where you simplify everything down to maybe something which doesn't even make sense at some point, which is, trust me, this works. You try to do something like this, and you estimate that, okay, this is a perfectly round cow, and now we can do something with it. Is this correct? No, definitely not. This is wrong. But the point of this is, if you make enough models which are wrong in different ways, then maybe you learn something more about the system that we are interested in, and in a fundamental manner. And that's the idea of these theoretical biological projects in sustainability, especially for agriculture. And let me give you like a small example of how this can be actually useful, right? I mean, is this kind of theory actually even useful for doing translational research then, in a way? Well, I mean, I would just put forth this quantity, and I think in the last 5-6 years, everyone, like epidemiologists, have been calculating this R0 for centuries, but all the way from epidemiologists to politicians, everyone was calculating R0s in the last few years. And that's because, and this quantity comes from an extremely simplified mathematical model, right, which makes a lot of simplifying assumptions, but it is still useful for us all the way towards policy making. And yeah, that's what I would say, that there is some value, let's say, in trying to do some theoretical biology. And I would just give you three short examples of how we can leverage it for actually sustainable agriculture, which I was very happy to hear about crop rotations already, which was mentioned before. And let me just start with that, in which how you can connect something which is known as the Parando's paradox to crop rotations. And Parando's paradox, again, coming from very fundamental concepts such as a Brownian ratchet. That's where it comes from. And the idea is, let me just put it directly in terms of agriculture, where now this is what I mean by making the cow a perfect sphere. You can just think of cover crops and cash crops. Cover crops increase the soil quality, and cash crops, well, they decrease the soil quality. You can sell them, make some profit, right? Well, you can also sell the cover crops, maybe with some probability, because you can have cover crops like cloves or so, which you can make some profit. You can let farm animals graze on it, that gives you some profit. This is say a probability of 0.4. You can make some profit out of it, right? But for cash crops, it's important that you check what the soil quality is. If the soil quality is above some threshold, then maybe you get some profit, but if it is below, maybe you don't make any profit, right? That's the probability of making a profit, 0, because your soil is not even good enough to grow the cash crop. Well, if it is good enough, you get some profit out of it, which is quite high, let's say. Well, it's not 100%, but so it's high enough. And that's kind of like the two simple scenarios where we have simplified a lot of things out, right? But this is how it looks like, right? So what's the frequency of cover crops? If it's 100% cover crops, then yeah, that should be cover crops. Yeah, that's right. So you make some profit, right? At least this 0.4. It's not even half. Right? So you're not even making profit which you would make by chance. It's less than that. It's less than 50%. If you put all cash crops, which would be frequency of cover crop 0, then you're making no profit. Because in the long run, the cash crops just deplete the soil quality, and there is no future in the long run if you're not planting any cash crops, any cover crops in between. So the profit is basically just 0. Now, those two are the ones what we call as the two losing games. So you lose if you play both of these games for a very long time, right? Only cash crops or only cover crops. Interesting things happen if you mix those two, right? And this is something that we do know. If you mix them in the right proportions, actually the probability of making a profit is more than by chance. This is an extremely simple model which actually comes from Harmer and Abbott kind of advertised it. It's called Parando's paradox, pushed by Juan Parando. And it's a idea which comes from Brownian ratchets. Extremely simple physical properties. And what this shows that if you have two losing games, you can just mix those two losing games, but you have to find the right proportion of mixing those two games. And that would mean we would need to know what is the right proportion of mixing cash crops and cover crops, so that we make, on an average, a profit in the long run. Now, those different lines that you see, well, they are not going all the way always above 0.5, right? And that's because what we also tried in this kind of work was like what happens if you include pathogens into the field, because you have crop pests, and crop pests make sure that you can't make actually a profit for that long. So how do we deal with that? That brings me to the second part, which is including classical theoretical ecology, Lotka-Volterra dynamics, and somehow connecting that to pest management. So now we include crop rotations and pest management into one single model. Again, looking at a simple scenario where you have soil quality, let's say on this y-axis, which increases when you put a cover crop, and so it decreases when you put a cash crop, right? The cash crop just uses it out, and so it goes down. But we also connect it to some kind of a cash yield. Maybe you don't make, here in this case, any profit when you plant a cover crop, but you get the profit when you have the cash crop. Extremely simple model, and on top what you see is a rotation pattern, right? That's the rotation pattern we would like to calculate in the end. So we went through the whole steps of calculating the optimal rotation patterns, all and everything, and what we want to connect it to is including these plant pathogen dynamics, and are those sequences that we calculated actually sustainable? And what we mean by sustainable is it in the long run, because what we want it to last for long, right? So in this case, for example, you can see you can go for 30 years, and you're getting quite a good cash yield in the end. The thing is, like, the cash yield increases every 10 years because the sequence is a periodic sequence of 10 years. So you can use the same sequence again and again and again. But the rate of increase of cash profit is actually decreasing. So you're not making the same profit every 10 years. And the reason for that is in the second plot, is because, okay, you see there is a slight brown line at the bottom, right? Which keeps on coming up, but then it goes down again. That's the pathogen. So the cover crops are good at keeping the pathogen down, but only for so long. And that's because, we come to the last plot, is because we include pathogen evolution. Because nature is not going to stay constant, again, as we all know, from the last few years. It is going to increase in virulence. So if the pathogen increases in virulence and adapts to the plant, at some point, at the end of 30 years, you can see the exponential growth again. So the pathogen will come back up. So there is some kind of sustainability in this kind of sequence, but still not for super long, right? So including pathogen evolution actually still breaks down the concept of long-term sustainability in this case, even for sequences for cash and cover crops. Which brings me to the last project, which was at least to demonstrate the idea, which comes from branching processes, which is like a classical mathematical process that one uses to calculate if things are going to increase forever or they are going to go extinct. You can write down very nice probabilities and times for extinction and things like that. But how do we connect it to something that we are interested in this case, which is perennial weed control and herbicide resistance evolution? So we came up with a very nice mathematical model based on those branching processes. But then we realized, okay, and this was focusing on Johnson grass, and what kind of control measures do we use to control this weed of Johnson grass? Well, typically people use a lot of herbicide, which we want to reduce, or there is a lot of tillage, again, what we mentioned, what we heard before, which we would prefer to reduce. So there is a high chance of failure of these control measures. And one of the big reasons for the failure is actually herbicide resistance evolution. Because we are dumping so much herbicide in there, at some point the plants are also getting resistant to the herbicide which is being dumped. And from all these studies, what we kind of see is that then is it even possible actually to control this kind of an evolutionary potential which exists in nature by default, right? And we are trying to kind of combat it with the brute force of, say, monocultures or other dumping heavy pesticides and stuff. So are there methods? Yes, I mean, there are clearly methods. Again, we have heard them throughout, which are about, say, for example, genetic control strategies or integrated pest management, where you can mix different kind of control methods like biocontrol together with tillage, and that might actually improve the adaptation of such kind of ideas, right? Because then you can demonstrate in the lab or in the field trials that, look, this might be possible of at least reducing the evolutionary potential or controlling it in the short term, as far as we can see. But the problem is, when you propose these kind of ideas, there is some kind of hesitancy, right? I mean, the amount of, the number of countries which are actually, say, for example, okay with having these genetically modified crops with increased resilience to be adopted. It's not very, say, immediately perceivable that, okay, this is going to work throughout, right? Most of the countries have not even made a decision about what they want to do with it. So this actually goes back to the question of resistance, but not in biology, but also social resistance, because the resistance is not just in these biological contexts of herbicide resistance or pathogen resistance evolution, but we are also very immune to changing our behaviors. And it takes time to adapt to, say, to new ideas or something, which, again, I would give an example of the adoption of these new rules, which everyone had to be somehow comply with for the pandemic. But it took there was clearly resistance in many places. So resistance to the adoption of new concepts, and that's actually something we tried to look at this problem again, and then you realize, okay, because this is not just in isolation, right? So this evolves in a social system. And that's one of the nice things about the different kinds of wrong models that we do. Because at some point, you can put these wrong models together in the right way to connect different things like epidemiology, economics, and evolution, which you see at the bottom. And then hopefully that gives you some kind of a better picture of what we would need to do when you integrate all these different aspects, which is then possible by mixing together these slightly simpler models. But that also brings me to the power of theoretical biology. As I mentioned, we can go across scales. And if we are looking at social systems, then we have to look at the right side, right, of this spectrum. And at the right side of the spectrum live all these kind of weird things. So we try to ask the question, like, what is the common thing between all these kind of weird things? And what is that there is a power of belief in here. All the concepts which are listed above, they exist because we choose to believe them in a social manner. They are social narratives, and their social narratives are very powerful driving force for people to come together and do something together, which exactly what we see, for example, in this room, right? That's what's happening. But narratives, they act as powerful coordination devices, and while they can bring people together to achieve these common things, they don't need to say anything about what exactly those narratives are. So what we again showed together working with cultural psychologists is that if you provide people a good narrative, then actually they will give up their selfish beliefs and actually can come together and trust each other to get something done together. And this was motivated by one of the very nice quotes by Daniel Kahneman, that no one made ever a decision because you give them a number. I mean, we have seen that before. You provide a lot of data to people. That might not be enough, so you provide a point. That's not telling people anything. How you get to that point, what's the story behind it, that's the thing that motivates. And this has been actually seen in, for example, climate mitigation problems. So, for example, if you want to win the popular imagination, you change the game, we need to tell new stories. You need to convince people that the new methods, for example, or the new techniques that we want to push are not just like, okay, here's the data that we show, that's why it will work. No, there is a deeper motivation behind it, and that would make it work. For example, that has also been shown for fisheries, that if you adopt this kind of a scenario, how the world will look like in the next 60 years. That's something that provides a much more positive, motivational story for people to adopt it, which is extremely necessary, I think. So what I would like to propose at the end over here is that when you look at agriculture, you can think of it as what Socrates put. It's like the best thing ever that happened to mankind. Or then, as we know a bit now later on, that of course it came with a lot of problems as well for the human species, with diseases and perdition. But whatever it is, agriculture definitely acts as a coordination device. And it is a persuasive technology. It changes our behaviors subtly. But now we see the effects quite drastically, but it has changed our behaviors over millennia. So we need to treat it as such and then use it as a psychological tool as well. Only then we will be able to drive this change. So with this, actually, I would like to say that achieving sustainable agriculture is a multi-scale issue, which I hope you all already know. That's not a surprise. But the thing is that the theoretical biology is also scale-free. We can go across these scales. And that's the nice thing about doing theoretical biology, and it can contribute a bit. And we can use it to promote more sustainable practices then. I would like to thank the program organizers. There was a lot of organizing thing, which I discussed a bit before, and my wonderful collaborators and funding. So thanks a lot.
Well, I don't know how the speakers were selected for this session, but whoever did the selection did an absolutely fantastic job, I have to say. Right, the final speech and talk of the session is from Abraham Dom, who's the chief scientist from Israel. And I'll turn the lectern over to him, and we return back to proteins.
Yes. Alternative proteins. Okay, thank you very much. And my talk is somehow related to what Amy started with the alternative proteins, but I will discuss the move from laboratory to the industrial scale of production of alternative proteins, mainly focusing on cell movement from the cell to the cultivated meat. So, okay. So the motivation to have alternative proteins from animals, okay, or replace animals, is the contribution of animals to, or ruminant animals, to the climate production of CO2. Animals produce methane, and methane is 80 times more toxic than CO2. So if we can reduce the use of animals for food, then we may reduce about 10% of the CO2 production. And this is just, for me, it is a side effect. The main effect is that I'm vegetarian and I prefer not to use animals. So how we produce alternative proteins? There are three ways how to do that. First is producing from plant, okay, take plant and or make the consistency in a way that it will look like a meat, and people will eat it, hopefully. Second is to use fermentation of bacteria or fungi to produce certain proteins, and then incorporate these proteins into food. And the last one is the one that is more challenging, is to take cells from an animal and grow the cells into a steak. So in the first technology, we have to select the right crops that the mixture of these crops will produce something that will look like or taste like meat or have the texture of meat. And here we have also to fit the proteins to the proteins of animals. The second option is, as I mentioned, to produce or to generate proteins by using yeast or algae or bacteria, and isolate these proteins and incorporate it into food. This is, of course, not so direct to a piece of meat, okay, or steak, but it is very useful for making food like milk and similar products where they are based on a specific protein that is needed. So, as I said, the main challenge is to take cells and from an animal, let's say a cow, and these cells to grow them outside of an animal, and eventually have a kind of meat. This can be either sheets of meat that later on we mix them together and prepare something that looks like a hamburger. Okay, it's not a steak exactly. And this is one way. And another way is to, or a more challenging way to make such a steak is to incorporate several types of tissues in one growing piece of meat, where you need, of course, the muscle, you need some oily type, and the taste should be similar to a steak. And as I say, there is a company that is trying to do that, but this is the most challenging in the field of taking a cell and grow it into cultivated meat. So the field of growing a cell into a piece of meat is actually started from basic research on tissue engineering. What we see in the left side, where tissue engineering, the concept of tissue engineering is to grow outside of the body a piece of tissue, and then use this piece of tissue and implement it or incorporate it or fuse it into the body where it is needed. For example, if a patient is missing an ear due to a car accident, then may manufacture the ear from the same cells of the same patient, and take this ear and stick it where the missing ear is there. And this is the challenge. Okay? Other challenges are preparing an artificial liver and the artificial internal organs that are functioning and producing what the body needs to be done. But this is a big challenge, because you can make everything perfect outside of the body or on cell culture, but when you implement it into a patient, then in most cases, even today, there is no success in the field of tissue engineering. Fortunately, the field of protein production, alternative protein came in, and then the people from the field of tissue engineering, including myself, switched into the production of cultivated meat, because the halfway of growing tissue outside of the body or in tissue culture, this was achieved already. So just taking it and moving it into cultivated meat, but the challenges are significant because for medicine, the cost is less of an issue. But for food, it is indeed an issue. Just to know that a piece of steak, okay, that companies are giving only to presidents or prime ministers to taste, okay, each bite probably costs $2,000. Okay? And so we are a long way to have a cultivated meat in the market. So we started with tissue engineering. Then we have to move into food engineering, because to make a food, it's not just to make a piece of something. Someone has to eat it and to feel it and to taste it and to have it as part of the culture or part of a food that is tasty. And then if we solve this, then we have to produce in large quantity. Okay, for the medical field, you need probably 100 years per year for the entire world. But a piece of meat, you need significant amount of tons per year. So the growing of tissue outside of the body, this is known, this is done already for many years in culture. And the way how it is done is cells do not grow in a two-dimensional way. This is only the skin. The skin can grow, and this actually was done many years ago, even 40 years ago. Skin tissue was grown, and actually it was a company that used to produce skin for people that were burned. But still, it was not that effective, so this company doesn't exist anymore. But the way how it is done to make other tissues is you need a scaffold, where the scaffold is a piece of plastic or a sponge that needs to be degraded as the growing tissue or growing cells propagate into this scaffold. So the scaffold starts for the food industry. It starts from soybean proteins, and then the cells are grown onto this scaffold, and eventually what we get is a combination of cells that are integrated within the scaffold, and this is actually the food that you are eating. So when you are eating cultivated meat, it still contains the sponge that the cells are growing on. Okay, and then so we passed already the basic science on it, and now we have to move from a small scale to a large scale. Here we have to select a certain type of cell, whatever we decided. It can be a chicken, although chicken is not an issue for the environment. The main issue is cows and animals that rumen and produce the methane, as I said. Then we have to have a big vat where we load the cells. The cells start to propagate, and then we add the scaffold. Scaffold can be a small or micron-sized particles that we add in, and then the cells stick to these micron-sized particles and propagate there. So what we are generating here is a micro amount of or microbeads of meat. Then we integrate them into a hamburger or something like this. So this is the second challenge, okay, to move from a small scale to a large scale. And actually, we don't have such instrumentation because we never needed in the world to produce cells or to grow cells to the amount that we need for cultivated meat. And this is another challenge that we are facing. Then we have this kind of meat, okay, or pieces of meat, or sheets of meat, and how we take these sheets of meat and make them into a product. So if we go to the right side, we take the 3D printing. So we take these micron size particles that are pieces of meat and print them into a kind of a steak where we can prepare different types of tissues and then mix them together by the 3D printing. Actually, there is an Israeli company that is doing that now, or taking in the middle, what you see, taking the sheets of these tissues and stick them together so they get a feeling of a piece of meat. Okay. So if we move to another alternative protein, is producing milk from fermentation. And this is an Israeli company that already is selling their product. What they do, they produce milk, contains about 40 different components. But 39 of them, you can add them into milk, but one component, which is the protein casein, you have to produce it in large amount because this is the main component that gives the feel of the milk. And this is produced by this company, by a fungi that is producing this protein, and this protein became the main component. Actually, this company moved from Israel to, I think, Holland, okay, because in Israel they didn't get an approval for distributing this milk, and in Europe they are probably more flexible, so they moved to company there, and I think that they are making hundred thousands of liters of this milk, and it is, I'm not sure if they started already to distribute it, but this is one of the objectives. Another issue that is more relevant to the kosher people, or the people that are using kosher meat or kosher food, and the halal, and even the Christian people that are not eating meat during 1 month in a year, so it might be of interest. And the question is this meat that we are making from a cell, it is a meat, it is vegetarian, it is what? Okay, and this is a question, because if you solve the question, Jews may eat a pork, okay? Because you can take a cell, you make a pork, and then you eat it, and even the halal people may eat it. So this is an issue that is still open, but since the industry is very dependent on this answer, okay, so the Israeli rabbinic chief rabbi already decided that this is a non-meat, but you cannot use it to eat pork, okay? So it is something in between, okay, because it's too complicated to decide, okay, that you can eat it. Okay, so if we look at what's going on in Israel now with the cultivated meat, we have many companies that just started in the past 5 years. The most successful one are from the universities, 2 from the Technion. The 2 from the Technion, the Aleph Farm, and the other one, I can't remember the name. Both of them started from scientists that worked heavily in the tissue engineering. Okay, one of them even came from MIT with knowledge of tissue engineering, and she switched into this, but she still continues in the tissue engineering, but to see a product, probably she will see a product more from these companies than from tissue engineering. There is a big investment by the government to promote this field, because the government feels that food is an area that we should invest, and since we have the knowledge, and many people are knowledgeable and are willing to expand on it. So we attract investments from the United States and other countries to promote this area. And as I said, why this is growing in Israel, because we have a strong academia in this field. The entrepreneurship, so any professor at the university is not looking only to publish in Nature, but also how to make a patent and an investment. And this is probably the reason that is propagating this field in Israel. So if we look at the number of companies, all of them started about 5 years ago, and we have in all three fields. We have cultivated meat. There are a few companies because this is the most complicated field, but more companies are in the food based on plants. Because you don't need the regulation for that is much less than the regulation for cell-based food, because this requires some like an FDA approval. And okay, I just yesterday morning I was waking up and I saw this from Forbes, and they try to kill this field. And what they say, okay, this, the perception of the people will be an issue here, and this reminds me the 40 years ago when the companies in the United States started to prepare crops from genetic engineering plants, and then the community said, oh, we cannot do that, and then the companies actually were not successful in having this product into the market, and this is an issue, because if you put in the perception of people that it is not good, it is causing cancer, okay, it doesn't matter if it is true or not, okay, but the FDA has to put his stamp on this technology, and I'm happy that the FDA already approved one product, and many other countries are moving into this direction, and hopefully the first issue is not an issue. Second issue is always an issue, patents and the companies that are fighting. If there is a success, okay, patents matter. If there is no success, no matter how many patents you have, okay? So this is an issue. And then the issue of does this save the world? Yes or no? This is business. Business can do good to the world, but the main purpose of a business is to make money. Okay. And if I can summarize about this field, it is the cultivated meat is saving 90%. Of land, because you can do everything in one big building, and you don't need the farms. Less water, because cows are drinking water for a period of time, and not all the water is consumed properly. The less air pollution. This is, of course. So from all of these parameters, there is a justification to continue and develop cultivated meat. Thank you very much.
We are running a bit over time, but I'm sure you can manage. And even though the session was about food, we are delaying you from getting to food. As our speakers gather here, so while you think about your questions, I'd like to use the chair's prerogative of launching a question of my own. And three of you raised the issue about community acceptance of some of the new technologies that we have. And in the agriculture and food sector, we have I suppose, the disastrous experience of genetically engineered crops, which have been almost universally adopted in some countries and almost universally denied and are stopped in other countries. And even after 30 years of safe use of that technology, it's still extremely problematic in many parts of the world, particularly Europe. Now, Amy, you particularly talked about some of the actions you've taken to work with the social scientists to address this. I wonder whether if you and the panel could sort of tease out a little bit more about how we in future can improve our interactions with the social scientists and make sure we don't face similar problems with some of these great technologies we heard about this morning.
Mm-hmm. It's an excellent point, and I think something we've thought a lot about the analogies to GMOs and sort of the public perceptions of that technology as well. I think part of the distrust about GMOs, for example, has come about with the domination of particular companies, for example, over these products. And so I think to combat this, I think that's where academic institutions and researchers in academia have an important role to play in communicating the launch of new technologies into the public sphere, particularly around novel foods. I think companies are clearly playing an important role in marketing as well and in communications, but I think if we can drive dialogue around these technologies, encouraging transparency around how the products are being developed and the impact that they might have, I think that's an important first step. I think with a deeper understanding of public perceptions and fears or challenges, that also helps us to better direct our communications and target, for example, in the case of cultured meat, a lot of people, consumers out there are very aware and recognize the impact of foods on the environment, and so if we can better understand the potential reductions in greenhouse gas emissions, for example, that we could achieve with cultured meat, that could be a way to kind of counter some of the negative perceptions and dialogue out there. But I think it's super critical for researchers and in academia to be able to contribute to that dialogue. So I'll turn it over to
Anyone else care to risk a comment on that? Avi?
Yes, I may add that perception of people, okay, you will have always. If you saw in the corona, okay, a significant, or I don't know if significant, but it was a voice not to use the vaccination by messenger RNA, okay? So those people will stay always. What we have to do is to leave this side or the people that think differently as isolated, okay? And actually there is a GMO already, GFI, that is doing an international effort to have the cultivated meat as part of our future. Okay, but you never know.
I can't see too well from here. Ah, yes. Yep, go for it.
I don't think we could have had better demonstration what yesterday the panel on the HO biology meant to say. Thank you so much for this wonderful presentation. So I have two questions, if I may. One is rather specific and technical, which goes to Gabriela and to Jean-François. How far do you think we are from the stage where we will be able to double the light use efficiency of the photosynthesis at a scale which would be applicable to the challenge? That's for you, Gabriela. And to you, why is this possibility not explicitly included in all these scenarios, which I think are very comprehensive? A second question goes to all of you. We seem to have two trends here. One trend is to do what we can to improve efficiencies and management, and etc., including the one on the, but still stay with nature, still stay with plant, and still stay with animal. The other one is mind-boggling, really. Cultivated meat, getting 90% plus reduction of all possible negative things. So which of those two trends will win? And the one on the meat cultivation reminds me other trends I know very well, like the hydrogen, for example, like the desalinization of the water. The only issue there was the cost. And in both cases, like the desal, at the moment the cost became lower for the general use, technology won at large scale. it will happen to hydrogen too. So what is the threshold in the cultivated meat? Is it a price only, except of the all the consumer preferences? So which trend will win, and when the breakpoint will come for the large-scale use of those technologies?
So
I guess
Gabriela.
I'll start in response to the portion of that. So what simulations suggest is that optimization of the specific aspect of photosynthesis that I talked about will give you a maximum of 30% increase in biomass. So the doubling, the 100% increase that you talked about, certainly can't come from one individual piece of that. And I think this just serves as an example of the need for fundamental studies of many different aspects. So I would say in many ways we're pretty close to the knowledge, but how that implementation will be achieved is still a really open question. If it's going to be achieved by looking at a variety of different strains, you know, sort of some of the incremental approaches that Jean-Francois talked about, through transgenic or GMO approaches, which have all the problems that we were just kind of brought up, I think that that's still an open question. So, you know, I think in some ways there's a lot of fundamental knowledge that could already be pushed forward, but it's really about that integration with policy and social sciences that is in many ways limiting the next step for increasing the global primary production.
Thank you. Well, thank you, Pavel, for your question, which is highly relevant, obviously. So as Gabriela explained, we have a perspective here to increase primary productivity in crops through such actions. I would like to cite work from a colleague, Stephen Long, from I think Ohio State in the US. And Stephen has been really leading efforts with GM modification of crops like soybean. And he achieved actually increases in crop yields in the field. And this was why, by working, I think it's quite complementary to the work from Gabriela, on the adaptation of plants to the rise in atmospheric CO2. So we do understand that plants were evolved actually in lower concentration in CO2 than today. So there is possibly a perspective of re-engineering photosynthesis to adapt to higher CO2 levels. And that has been achieved in the field with around 20% increase in grain yield. Same for other works with actually adaptation to oscillating light levels, which were proven to increase the grain yield. So this is really relevant, and I'm with you when you say I should do some further efforts to include such innovation pathways in the sort of modeling we are developing. Yes, this is a very nice perspective, and we should do this. However, if I take a step back and come back to what you said, Peter, about these problems with GM we faced in Europe at least, and I think it's also true with other countries, we could also relate it to your work, which is quite fascinating, because in some sense what the big players in the seed and herbicide markets did is to create a miracle solution, you know, a plant that would survive a herbicide that they were selling. So for the value of those companies, it has been a huge, obviously, benefit. But for the consumer, given exactly what you explained with the evolutionary games, which means that you're not always going to win with the weeds, because they will in fact adapt to your herbicide, this means that we have created huge problems. There are many countries where we are now using herbicides with no more efficiency because they are herbicide-tolerant weeds that are just surviving everything. So you know, it's an interesting example of an innovation which was of huge benefit for companies, but possibly not solving the problems to the scale needed. You know, so I'm not a specialist at all about cultured meat, but I must say I'm a bit skeptical because when we saw your figure about the 92% decrease in greenhouse gas emissions, I would like to challenge this because there has been a number of papers showing that if you take a life cycle approach, you do use energy, you do use some nutrients, some chemicals, and so on. So in a life cycle approach, we're not going to reduce as much the greenhouse gas emissions. However, it could be you could reduce it, but not to this large extent. So let's always try to have a balanced view about our innovation and the long-term impacts of the innovation. Otherwise, there will be a backslash on innovation. Please realize that a number of people everywhere in the world are really skeptical about the potential of technologies because of those past backslashes. So we need to really work with everybody to create sound innovation.
Avi, you wanted to comment on this as well?
So I'll comment on, first of all, I agree with you 100%, okay? And you know, the people say 15% reduction if we get rid of all the ruminant animals, but I doubt if this will be the case, because I believe that only 10% or 20% of the people will switch from real meat to the cultivated meat. Okay, so this is one issue. Okay, but still, from business side, you have to say otherwise. Yeah. Okay. The second issue of the cost of producing the cultivated meat, the main cost are the growth factors. There are 4 growth factors that are used, and they, I think it's 90% of the cost is just the growth factors. So what we try to do is to find alternative for the growth factors, or a way how to make them much cheaper than what we do today. Because today we take growth factors that are used for human therapy and use it. Because of that, it is highly expensive. But if you can find a way how to make it in a cheaper way, then you solve this issue.
Melanie, I think you've got a question.
Thank you. So I'm going to raise the level at which we're looking at this. So obviously, this is a very complex system with many different factors. And if we really want to achieve sustainable human nutrition and a healthy planet, I'd be interested to hear from all of from the speakers this morning about what one shift you think we could do now that would make the biggest difference. And I know that's a challenge because it's a complex system and it's putting you on the spot a little bit, but I'd just be really interested in those perspectives. Thank you.
Right. So we'll start with you, Amy, at that end of the table, and your answer to that question and work our way through.
One shift right now, I mean, I think the regenerative agriculture approaches that we heard about have a lot of potential. They're already being implemented on smaller scales, and that, I think, has lots of environmental benefits and also human health benefits. I mean, clearly into the future, I think clearly novel foods, I think, can play a role as well, but we need a diversity in approaches, I think. But since you asked right now, that would be my answer.
Thanks. Gabriella?
I'm going to say moving towards more widespread testing and investigation of transgenic or GMO lines for increased biomass.
Okay, that was an interesting one. Peter?
Well, to return to that earlier discussion about genetic engineering, I think the messaging is probably a little skew on that. I think there are pluses and minuses, and I think the public tend to respond more to negative news. And genetic engineering isn't one thing. There are uses and misuses of the technology, and it was mentioned that people are slow to pick up technology, but they're not really. It depends on how attractive that is. And so I think if the messaging around the positive aspects of genetic engineering is tweaked, I think that will make an instant difference to how that technology is applied and how soon we can benefit from it.
Okay. Jean Sfra, what would you go for? Everything.
Yeah, my plea is really about a systems approach, connecting different things, so it's a bit difficult to answer. But, I'd just like to say a few things. One is, if you just look at, in a classical sense, food security, which is like a number of people that go hungry, then it's really a food access problem. It's not a matter of increasing production. We have enough production. And so it's far more about everything about food insecurity, conflicts, wars, climate change impacts, and so on. If you think about huge impacts on health that we have with malnutrition, chronic diseases, please rebalance your diets. We could do it. I mean, public policies are amazingly weak on this. There should be strong messages about rebalancing our diets. If it's about then how can we, in the long run, have sustainable food systems, please consider soils. Soils are really depleted in soil carbons. They are unhealthy. Even in Europe, the Joint Research Center from the European Commission shows that 60 to 70% of our soils are unhealthy. Well, unhealthy is a bit vague, but there are 8 criteria. I don't have time to detail. And lack of soil carbon is one of them. So, answering different problems, prioritize things in different ways. That's possibly my message. I agree with regenerative agriculture, if you like to call it in this way. Protecting soils and, using a bit less inputs would be beneficial.
Yeah, I actually remember there was an old analysis from the FAO estimated that less than 20% of the world's agricultural land soils are in a healthy state. 80% are severely degraded. So, next.
Yeah, I mean, as we have been hearing, I mean, there are so many fronts that people can work on for this, and people are working on, and definitely improving crops from using different techniques, from genetic techniques, or combating pests and pathogens, also using those techniques, but putting them together with what is well known, like something which go out of fashion, like biocontrol methods. So there are different ways in what one can go forth with that, but also something that I saw a picture of, but we didn't discuss much about, is also controlling food waste, because exactly as you mentioned, like there is a lot of production, but a lot of it also is going waste. So something in that direction. But to cover all of that and give one answer would be actually to somehow be able to fight misinformation and then lack of information. So I mean, controlling for both of that. So controlling the misinformation which is there and providing more information. I think that would go a long way in promoting all these different techniques.
Difficult one. And Avi?
They say everything, so I have to be something different.
Something new now.
Yes, I would say that better fertilizers that are more friendly and more effective. And I know that there is a project in the United States that the VAX program to exactly deal with this issue. And I think that this is a major issue.
Yeah, there's certainly a lot, I think, happening in changing the formulation of fertilizers so they are more effective.
So are there any other questions out there? Yep, there's a hand waving here.
There's a light over there too. I can't see who.
Yes.
Thank you. My question is to Gabriella. In the 2 mechanisms that for heat regulation that you have nicely demonstrated, are these 2 mechanisms, maybe my description is a bit naive, are they complementary, or are they maybe a form of redundancy for the trees? Thank you.
Yeah, thanks for your question. So they're complementary. Photosynthesis, like all biological systems, is mathematically speaking a stiff problem, right? So there's dynamics on a lot of different time scales. So those 2 different mechanisms give responses on 2 different time scales, essentially, a faster and a slower one.
Okay, any others out there? Where are you pointing?
The light's going to come on and then I can't see.
I wish yes, thank you. I wish we had a chance to get a stronger input from humanities, like psychology or sociology. I'm a happy father of kids. I try to convince to eat broccolis and endives and tofu, and now we're talking about having a substantial fraction of the humankind changing its taste and eating some kind of goo that it never had any tradition for. So I think we really need to figure out how we can change people's taste in a reasonable time. I'm not talking about a generation time scale. It's much shorter than this.
Yeah, a good question, but probably one that's going to be very hard to answer. I don't know any of the panel would like to. How do we actually tackle this?
I can jump in. I'll take a stab. Yep. I think it's an excellent question. I think that really drives a lot of our work in generating novel foods that simulate and are mimicking foods that we already know and love, like the milks and meats. Achieving behavioral changes and changing people's diets is something, there's lots of campaigns you can point to around the world, but those kinds of behavioral changes are really hard to achieve, especially around food, where it's so deeply rooted in our cultures and evolution, even to eat high-protein foods. So I think that's where this first generation of novel foods is heading, right? To just mimic foods that we already know, so that it's not like goop. It's actually something that we recognize as a piece of meat. Into the future, I think there's really exciting potential to start to think about more precision foods, or tailoring foods to people's specific tastes or nutritional needs. That's sort of another level of that goes beyond probably the scope of our discussion today, but I'll turn it over to any other panel members.
The food industry doesn't have a great reputation, and pumping up sugar levels, fat levels, and salt levels have been the key new additives.
If you can now make meat that has, for example, a more healthful profile of fatty acids or higher protein content, or doesn't have heme, which is one of the putative causes of associations with colon cancer, I think we can imagine engineering more healthful foods that, aren't just engineered for maximum flavor.
Jean-François.
Yes, thank you. No, there is certainly a lot to do in terms of innovation. We have in France a challenge about innovation on fermented food. And fermented food is actually anchored in a number of traditional food cultures. With the tofu, for instance, but please consider the cheeses in France. We are quite proud of our cheese, and so on. And certainly, fermented food has a lot of potential, also with a rather sound environmental profile in many cases, to make a difference in terms of innovation in the food systems. We have also a fully renewed understanding of the health impacts of food through the interrelation with the human gut microbiota. And that's a fantastic, I think, perspective in integrated biology. A number of the chronic diseases appear to be correlated with shifts in the human gut microbiota. And I think if we consider also some of the past efforts to produce highly processed food, we now realize that some of the chemical additives in those food are impacting negatively the human gut microbiota, therefore creating some imbalances that could lead to chronic diseases. So this is also another pathway to link understanding in terms of human biology with the microbiology and also with the food innovation.
Thanks. Chaitanya.
Right. I mean, I'm not very, very qualified to answer the question about the behavioral changes, but I think that's just the result of me hanging around with way too many behavioral psychologists. But what I learned from them was also like the stories about the culture, about food, that we tell ourselves are really strong because we tell ourselves, a lot of them. If you look at the history of food from these different cultures, it has changed so fast. I mean, associating tea with UK, chili food with India, chocolates in Switzerland, I mean, where they came from after the Industrial Revolution, I think that's something which changed very fast. And I think that it is possible to make these changes also. It's, again, going back to like how we incorporate these new concepts into the culture and how we put them in. I think that would make a difference. But it is possible that behavioral change does happen fast enough. You don't have to wait generations then.
Okay, that was a difficult question, but interesting to hear the different perspectives on it. I'm going to use my prerogative as well. I was struck in both Gabriella and Peter's talks about the fact that there was lots of diversity still in the living system. So Gabriella, you showed us the differences between Arabidopsis, moss, and your alga. Peter, you talked about plants at different rainfall levels having different responses to water use efficiency. I'm wondering whether you think, in amidst all of this very high-tech approach, there's still diversity out there in the natural systems that we haven't explored that might actually help us tackle some of these problems.
Absolutely. You know, one of the strengths and weaknesses of biology is that we end up with a few model systems that we develop really robust tools to look at. You know, E. coli or Arabidopsis, which is the E. coli of plant genetics. And so that's a strength, the ability to kind of handle those proteins, manipulate those proteins in cells, but it's also a weakness because, you know, the ecosystem is incredibly diverse. And so evolution has really potentially identified unique solutions that we could certainly explore. And I think, you know, some of the people here today have explored them, for example, in cyanobacteria.
Well, I would say yes, the biodiversity, again, we need to probably think about how to use that biodiversity a little differently. Thinking about my own work, some of the problems we've had, even that example I talked about in parameterizing those models, it was kind of seen as an enormous task because at some level all plants are different, all species are different, but if we can find common threads through that biodiversity that they all share, I think that's what we need to understand a little better as to what are the commonalities amongst the biodiversity that we can use to simplify our models and our systems to make them more powerful and more general while accommodating that biodiversity. I mean, the photosynthetic process, all leaves have photosynthesis. At some level, there are these common threads that run through that biodiversity, but we need to discover them and utilize them a little better than what we're doing.
Okay, thank you. I think that's a really nice way actually to wrap up, to think about the commonality of process, the differences there are to find, and the way we bring that together with these novel approaches. So I'd like to ask everybody to join us in thanking our 6 speakers for a really interesting, engaging morning. And Guntram tells me that the good news is that we still have a full hour for lunch, and we'll start again at 1:15 sharp.
And speakers who haven't uploaded their talks for this afternoon, please go to the speakers' corners now, please. Thank you.
Okay, thank you.
So welcome back, everyone. I'm Ted Hewitt from Canada on behalf of my co-chair, Marina Picciotto. I want to welcome you to the next segment of talks this afternoon, essentially mirroring what we started on Tuesday, but now in expanded format. So I'm a sociologist, not a scientist by trade, some would say. One of the things that I've noticed in the sessions so far, and in fact throughout the conference, is that human and social actors, when they're portrayed and when they're discussed, quite often are essentially viewed as objects, objects for information to be transmitted to, or to be transformed to accommodate certain views or certain perspectives that would make for a better planet, absolutely. So we're going to shift now to a more subjective view of humans, as you'll see, more as interpreters or actors in the environmental context. And I think this is why we speak of the need for more social sciences, more humanities in the work that we do collectively, because at the end of the day, humans sit at the center of everything, at the beginning, at the middle, and at the end. So I think we're ready to go. We're going to split, Marina and I are going to split our chairing duties, so I'm going to introduce the first set, and at the end we'll have time for questions, absolutely, with the panel. So we're going to open with the first keynote, Neurophysiological Mechanisms of Social Interaction-Driven Language Development, and that will be delivered by Tomomi Shumogori from Japan. So Tomomi, if you're ready to go, we're ready to have you. Thank you.
All right. Well, thank you very much for the introduction. So I'm really glad to be here to introduce about our story. So what we are doing, what I want to talk about today is about the social interaction, language, and the brain. Okay. And I have a pointer, I believe. Okay, fine. All right, so when we talk about the social interaction or social communication, you can easily imagine how different animals are doing that. Like they smell each other, they use odor, and they touch each other and using perception. And also the other stuff like they use the pheromone, for instance, to interact or attract each other. But what is so different for human is for us, we use language to understand each other for the social communication. And you already know that, how much language means to us, because it has a lot of information. It actually provides a lot of information, and it helps to understand each other. So then in that case, what the language means to us and what exactly it is. Actually, it is a big question, I think. So since it's a big question, there's so many different fields that works on language. For instance, like a linguistic point of view, they think that this is the fundamental behavior that makes it human as human. So it is really important. For it in the field of humanities, they actually put the language on the top of the pyramid for all the perceptive, or attention, memory, motor skills, and all of those behavior. So this means that they also take language as the most complicated behavior to understand. And this is the reason why it is taking so long, and still we haven't solved all the problem to understand what the language is, although we know it is really important. From the neuroscience point of view, we actually know that which part of the brain we are using when we are processing language. Using high technology like fMRI and PET imaging, now we know that these regions are actually involved for the language processing, like a Wernicke's area or Broca's area. So for this we know, okay, there are certain brain regions that we are using to process the language, but still we don't know how we are doing that. And there's one interesting research done is like, what about those people who can speak multiple languages, how they are processing language and which brain region they are using while they're doing it. And then what is interesting is comparing monolingual people, they basically have all of these area, which we all know for the language processing, but they use those regions in a very different way. So while they're speaking, they basically, all of these regions are highly activated, which means that all of these regions are heavily connected. On the other hand, the monolingual people, they do have these regions, but they're only using certain region while they are speaking. So it seems that the neural connectivity is making the differences for these kind of different behavior. So this is also what we know from the neuroscience field, but again, we don't know how they actually get this connectivity while they are developing. Because we all know that acquiring language is not genetic. If you grow up in a different country, you become fluent with those languages spoken in that region, and even this language is not spoken by your parents. So what's happening while they are learning the language is also one of the mysteries that we haven't solved yet. But when we talk about language, of course, we need to look at human, because human's the only animal we know that speaks language like this. And what we know about the language development in human is they take stepwise development. Like starting from 2 months, they do a lot of cooling, and 4 months old, there's a lot of bubbling happens, and at 6 months, they start to distinguish lots of different sounds like M and Ds. But this period, 4 months old, bubbling seems to be a really important step for a human to learn language. So I will show you the movie that how infant and mother interact. So there's a sound comes up. Oh, wow. So I think you have seen this, right? Or you have done this to your kid. So what is happening here is basically the mother or adult talk to baby, and then the baby tried to reply with mimicking the sound. And this interaction is really important for baby to learn language, because if they don't receive that much input from adult, can I go back? There we go. From a mother, then they have lower number of the vocabulary after they grow up. Okay, so then how are we going to tackle this question down without using human? Of course, the human, there's a lot of ethic problem. We can't do everything like what we want to do. So the first thing is come up to your mind is, what about mice? Can we use rodent, which is the most frequently used animal in the lab? And as you're hearing, they do have different type of squeaking. So it's showing a different strains, and they do have their distinctive call pattern. Also for the baby mice, they do generate ultrasonic vocalization that we cannot hear, but it is important for a mother to retrieve them. But the problem here is these guys, they don't learn how to make this sound. It is innate, they already have it. So this cannot be the good animal model to answer what we want to do. Then, here we go. We were looking for suitable model animal, and now this animal is becoming, very popular among the neuroscientists especially. And this is a common marmoset, little New World monkey that live in Brazilian region. And I'll show you, or make you to hear that, what kind of vocalization they do, and then they do the vocal exchange, which you're going to hear it in this movie. Okay, so this guy is making sound. Now you can hear someone else is answering to his call. One more, I will come back one more time. I hope you heard the last bit. But basically, they wait for someone to make a call, and then after they finish it, they reply. So this is basically, it's not in the screen, but the animal is really far away. So this is a fecal they exchange when they can't see their friend, because it's a high pitch, and then they can actually hear it even they're far away. And there are only a couple of type of calls in this movie, but actually they do have a lot of different call types that you can see here. And also these different type of call seems to have a meaning like this. And what is also interesting about these animals, they can combine different type of call types, and then they can generate another meaning. Okay, so what is also interesting about the common marmoset vocalization is they need to learn from their parents how to make a correct vocalization pattern. So you can see here, this is the neonate, 1 day old neonate marmoset, which is a sonogram for their call. And then you can find that this typical yellow coal type, which this is a cry coal, is very typical to baby marmoset. So you can also see it at 14 days old marmoset, these yellow greenish color. However, if you wait for them to grow to 1 month or 2 month old, these yellow coal, the cry coals are almost gone. And these coal types actually transform to this long coal, which is a fecal we just heard in that movie. And that's the kind of adult coal type that they really need to communicate each other. And the reason why I'm saying that they need to learn from how to make a coal from their parents is because of this. We actually do have a big, marmoset colony, but sometimes there's accident, the parents actually neglect and they don't raise. And then in those cases, we, human, raise them. And then those baby marmoset, which was artificially raised, actually show interesting coal type, which is here. So this is 2-month-old marmoset, which the human raised, and you can still see lots of cry coal remain in their coal pattern. However, this is actually the sibling of this one, and there are no cry coals, but it already transformed to the fecal adult coal type. And I hope you can hear it. So this is the movie from this guy, and then once we put them back into its home cage, the sound is a little bit small. Okay, probably you didn't hear it. I was trying to show you or make you to hear that it is making lots of, cry coal once we put them back into the cage. So it's basically, he got back in his cage, so he got highly excited, so he's quite happy. But normally, baby marmoset don't use cry coal when they're happy. So they're using it in a very different or wrong way. Okay? So then, it was a simple, experiment, just show the one coal type, which is exist or not. But in the reality, they actually do make more complicated coal type. So this is also not that long, but if you look at this coal pattern, it's more noisy, right? But this is a coal pattern when they make it when they are separated from the parent. So when they're separated, they make a lot of coal type and try to call the mother or father for them to come and pick it up. In that case, we actually make this, complicated coal. Actually, this is after they call the parents, and parents actually came and saw them, but they didn't pick it up. So they're a little bit stressed. In those cases, they make this cry coal, thick, and trill. So it's a combination of this. But if you leave them a lot longer, so they are left alone, and then you leave them alone for a lot longer, and then they change their coal type. So now it is more about the cry and twitter. So this is basically parents came, but she didn't pick me up, and then for a little while, you have your kid in the kindergarten, sometime you're late, right? In those cases, they're probably, really sad, but it's the baby cry in a very different way. So probably this is what is happening. So you can see that, I hope you can see that, they do make a different kind of combination of the coal, which again means something. So here, this is really interesting, phenomenon that we can actually work on. And also, marmoset, it's not only because they are vocalizing animal, it is also, very beneficial for us because we can generate the transgenic marmoset or knockout marmoset using CRISPR-Cas9 technology. And then actually we are making lots of Parkinson's disease and also a developmental disorder model animal. And one of this developmental disorder actually have the altered vocalization pattern, which I'm going to show you. So one of the mutant line that we actually have right now is Rett syndrome model. So Rett syndrome is known as the disease that it happened mainly in girls because the causing gene MECP2 is actually on the X chromosome. So in the case of the MECP2 mutation happens, there are a lot of behavior defect, like a very interesting motor defect, like they just move their hands like this. But what is interesting for us is basically they lose language ability. Okay, so then we generated MECP2 knockout mutant marmoset, thinking this is could be the Rett syndrome model, which is we are still also working to make sure what is the similarity with human Rett syndrome and then also differences. But one thing we realized that the MECP2 knockout marmoset also have altered language developmental pattern that you can see here. So the left side is the wild type, right side is the mutant, and these are the time course that you can see that this yellow bar is actually the Twitter call, and it seems that they have less number of the Twitter in this animal. Okay, so there are differences that we have noticed, but to really understand what's happening is since they have a very complicated, let's say, grammar, and then different call type, and the combination of the call type, just chop out, small or short period, it doesn't make sense. We really have to cover the entire, time when they are growing, and also we also need to understand or see that how they are interacting with their parents. So we decided to generate this device that we can monitor and record the entire family for 24 hours. So now you can see here, there's only one monkey inside here, but it's basically keep moving around, and it's gonna make fecal somewhere, probably soon. And we are basically monitoring that, there you go, it made a fecal. We are monitoring the entire behavior and when exactly or where exactly they made the call. And then we can extract like which call type it generated, and then we can find out when and where they did it and then what they were doing. And also what we can do is after extracting, we can also extract only one call, like this is the extraction of the fecal, and you can find out where also they generated fecal. Now it's feeding a lot. Okay. So this is kind of the device that we have generated. We're trying to collect lots of data. So this is something we can do by ourselves. But what is really difficult to do is the analysis of all of these huge amount of the data. We need to understand, but it's of course just looking at the sonogram doesn't help much. So now we need a collaboration with the people who can model this pattern. So here, sorry, it's a really busy slide, but this is basically what we are trying to do here is we just get all of those call pattern. We can get interaction with their parents, and also we take all the pattern while the development. And then now we can, using the modeling system, probably can find out what's the rule for the marmoset to learn language, or what's how they usually interact with each other, what kind of input they need when they are developing. So this is the plan here. And also to understand how human vocalization develops. We can't just keep using or working on marmoset. Now we need someone who works in human baby. So here's the plan. We also get a lot of human baby vocalization. And also we have to take a lot of mother-infant interaction. And also what we want to do is, well, we can't do that much for human, but again, if we make them to hear a slightly different tone, how much ability they have to mimic that tone. So we can do those kind of things, and altogether with the marmoset data and human data, probably it will give us the platform that we can directly compare marmoset vocalization development and human vocalization development. And as a neuroscientist, we actually want to know what is happening inside of the brain. And once again, we have monkey and human. We can't directly compare because anatomically they're a little bit different. To solve that problem, now we have a person who can actually compare, sorry about the residual Japanese here, but this is the human, Diemen, and then this is the macaque and marmoset. So actually he is developing that we can actually compare these brains and find out same region or equivalent region for each of the brain. And at the same time, if we can get like FNIRS data for a baby, human baby, then we can apply to these information and can compare where exactly the brain is functioning while they are learning language. So this is basically the plan, but once again, it is not easy to do this everything by ourselves, so that's the reason why we have this team. And after we collect lots of the data set from marmoset and also from the human, there's a modeling system comes in here, so we can actually compare, it's an integration of human and marmoset data here, so we can get what's the similarity, what's the fundamental rule that actually governing the vocal development. And also we can do the cross-species imaging, so we can compare the brain region that we are using each other directly. So this is a really important part for the integration of different research area. Also, doing this, integrating lots of different research area, what we can do is something like this. This is just an example, but now we have a molecular biologist, and then also we can work on a lot of molecular, new advanced molecular technology to find out in marmoset brain, which may be able to translate in human brain. Also with this, we can deal with the language evolution or robotics, and also like predict the operation dynamics of human vocal learning. This is basically our ultimate goal. But with the interaction with all of these teams, that you can finally get this thing done. And we thought that this is something we have to deal with till quite recently, but again, this is like, they have to have the input and they start to learn how to speak in the proper way. But now we have to deal with some other input, which is the AI. So now AI can talk to you. I mean, you can ask something and they can talk back to you. And we know that when we are learning language, if you, watch TV, like a Spanish TV, you can't learn Spanish. But if you have Spanish-speaking nanny, normally kids become very fluent. So it's very different from just you're hearing the language or you have vocal exchange. It makes a big difference. But now, ChatGPT, if it starts talking, they can talk back to you. They can have conversation. But we don't know how much these things change our kids' speaking or behavior. So there's another factor now we have to consider about it. This is something that I wanted to add here, which I forgot. But this is, again, this is another integration that we need to consider about. And also this is a reason why we really need to understand how brain functions and how brain develops. Okay? All right. So altogether, these are the people that actually worked on this the project. Well, they're still working on it, and these are the collaborators. So these are all from this project is running by the entire RIKEN project. So these are the people from RIKEN and also funding from RIKEN. So I try to keep it short. Thank you very much for your attention.
Thank you, Tomomi. So we'll move to our next speaker in this first series, and that is Ariana Strandberg-Peschken from the Max Planck Institute for Animal Behavior in Germany, and she'll talk about communication and group coordination in animal societies. So, are you all set? Fantastic.
So thanks so much to the organizers of this conference. It's been a really fantastic time so far, and I've learned so much from all these different interdisciplinary perspectives. So it's really been a lot of fun. And how do I, let's see. This one, forward. Aha. So my research centers around the topic of social behavior, and I think as humans, we can all really relate to this topic. People live in interconnected societies, and we're intricately dependent on our social connections and indeed on our broader scale societal structures to function and to interact with every day and potentially even to help us survive. But we, of course, are not alone in being so social. Sociality is something that has evolved multiple times across multiple different animal lineages and comes in a variety of different shapes and sizes, forms, and different contexts in which social behavior is observed. So why is it that sociality is such a widespread phenomenon? Well, there are a number of different benefits to living in groups. For example, being in a group can enable group members to better be able to find resources in their environment. It can allow them to avoid predators, either by having multiple group members looking out for danger, or by having rapid responses when predators do strike. And finally, being in a group can also potentially enable us to enable animals and humans to make better decisions. For example, by integrating information from multiple perspectives, like we're doing in this conference, and also by taking advantage of canceling out biases in phenomena like the wisdom of crowds. But in order to reap these benefits of group living, animal groups need to coordinate with one another. And they do that in a wide variety of different contexts. For example, many groups of animals move around together throughout their territory. And this means that they have to be constantly coming to consensus as a group on where and when they're going to move. Coordination is also required for initiating changes in group activity state. So for example, going from a group resting state to a group traveling state. And finally, coordination and cooperation are needed to join forces to face common threats. For example, mobbing of predators or defending groups against neighboring competing groups. So underlying all forms of coordination is some form of communication, be that visual, olfactory, or acoustic. And in my research, I focus primarily on vocal communication and how it underlies coordination processes. So vocal communication can be important in all three of these different contexts. Many animals have what are known as contact calls or cohesion calls that allow them to kind of ping back and forth while they're on the move to stay in touch with their group members, especially important when they can't always see each other. Vocalizations are often also involved with initiating group departures. as well as for recruiting other group members to join in collective action and for coordinating those actions once they do happen. And so my research centers on this topic, this interaction between coordination and vocal communication. And broadly speaking, we're asking the question, how do groups achieve coordination? And what role does vocal communication play in driving these coordination processes? But this is something that's actually relatively difficult to study, especially in natural social groups. And the reason is that our traditional ways of studying behavioral biology in the field, which are extremely useful for looking at social behavior in general, involve humans going out and observing animals directly. And although these methods can and have provided so many insights into diverse social structures, they're not particularly well suited to looking at these coordination processes because basically you have many individuals interacting across a wide spatial scale sometimes all at once. And so I think this challenge is nicely put in this quote from a chapter on baboon leadership, actually, by behavioral biologist Richard Byrne, when he said, "It's an observational task of daunting dimensions to attempt to record the actions of many potential decision-maker animals at once." So to tackle this daunting task, we have a lot of really exciting new tools that are helping us. And one of the really ones that I'm most excited about is called biologging. So basically, this is when you have small tags that you can deploy on animals, either attached as collars or backpacks or some other type of attachment. And these tags contain sensors that can tell you something about the behavior and movement of the animal. So often tags will contain GPS loggers, which will tell us where the animals go. They can also contain accelerometers, like you have in your phone, which can tell you, how the animal is moving and potentially what behavior is, as well as a variety of other types of sensors, like microphones, temperature loggers, pressure loggers, and so forth. And so these new tools are really opening up exciting new directions across the whole range of behavioral ecology and movement ecology, from looking at local scale social interactions in groups to mapping broad scale migrations across the globe. So in my work, we're using these tools to look at these coordination processes and how communication is playing a role. And we've started a interdisciplinary and international collaborative projects funded by HFSP, where we're called the Communication and Coordination Across Scales project. And in this project, we're bringing together an interdisciplinary team of field biologists and computational scientists to look at the question, how do social animals use vocal communication to coordinate group behaviors? And we're investigating this question across different species, across different spatial and temporal scales at which they're coordinating, and across different contexts. So in particular, we're focusing here on 3 different species of social mammals, and we see these species as spanning kind of a range of different levels of group cohesion, and therefore a range of different types of coordination that we see. So on the left-hand side, you have meerkats, which are form highly cohesive groups that move around together throughout the day. Basically, they have to constantly come to consensus on where to go. On the other end of the spectrum, you have spotted hyenas, which live in very dispersed groups where all group members know each other, but they're not all together at once. They form small subgroups or move around independently, and those groups split and merge over time. And then in the middle, we have coatis, which are kind of in between and do mostly stay together, but also do some splitting. And so we're using these 3 species as kind of a test bed for looking at how these species solve an overlapping but also somewhat distinct set of coordination challenges using vocal communication. Today, due to time, I'm just going to tell you a little bit about the meerkats, although I will show a little bit on the coatis and hyenas at the end. So just a quick primer on meerkats. They are a social mongoose species that live in groups of anywhere from a few to up to 40 individuals. Like I mentioned before, they travel together as a group while foraging, and they forage by digging in the ground. So that means that they're often kind of not looking at each other, but rather looking at what they're doing. And because of that, they use a lot of vocalizations to coordinate movement. Because of a long-term study called the Kalahari Meerkat Project, which is run by my collaborator Marta Manzer, we actually know a huge amount about the communication system of these meerkats. We know, for example, that they have over 30 different types of vocalizations that have been characterized, and that they use them to coordinate a variety of different social behaviors, many of which are involved in these collective movement processes. So if you think about this kind of coordination challenge and bring it down to the level of the individual, that individual at any moment in time has to be deciding where it's going to move, and it might do that as a function of information that it can see about its group members and relative spatial positions, as well as the vocalizations that its group members are giving. And at the same time, that individual also has to be deciding what it's going to say, how it's going to call, what types and what rates of vocalizations. And it might do those things also based on those two factors. And so what we have in these groups is this complex interplay between movement decisions and communication decisions that we need to unravel to be able to understand how this coordination process is working. Because it is the combined actions of all the individuals in the group that is leading to the ability of the group to maintain cohesion and to decide where to go. And this brings us back to that observational task of daunting dimensions, because it's very difficult to monitor, to know what everybody in a group is doing at once. And so we followed the suggestion of Richard Byrne in this project, and we attempted to monitor the decisions of many potential decision makers at once. So we did this by using those biologging tools that I mentioned at the beginning. So we put small tags on these meerkats. Every individual in the group gets a tag that has a GPS device, which allows us to tell where that meerkat is every second, an audio recorder, which tells us what that meerkat is saying to its neighboring group members, and an acceleration logger, which can tell us something about its behavior. And so when we manage to deploy these collars on groups, what we get out is data that looks like this. So here, each dot represents an individual meerkat. And the little symbols that hopefully you can see coming up around the dots represent the vocalizations that those meerkats are giving. So for example, you see probably a lot of these sort of gray circles coming up around the meerkats. Those are those cohesion calls that I was mentioning in the very beginning, where the meerkats are using them to kind of ping back and forth to keep tabs on one another. We also know that these cohesion calls are attractive to the meerkats. So basically what we think is happening is that they are, and they also stimulate the production of other meerkats to make those calls. So what we think is happening is that they're sort of using positive feedback to make a kind of hotspot of calling, and that this hotspot is allowing them to coordinate movement. You may have also noticed, if you were watching carefully, that there are lots of other different types of calls. In particular, there was one individual that started giving an alarm call, and then everybody ran. So we can also look at these moments of potential danger and see how the group manages to respond to them. So there's a lot to dig into with this data. We're also developing a lot of AI tools to try and both extract the calls and to make meaning of the data and build probabilistic models at the individual and group levels. So, but of course, these social interactions are not happening in a vacuum. They are happening in a real environment, and this environment is going to affect the communication channels that are available. So one really important question for us to address when thinking about social species is, how do these coordination systems respond to environmental change? And to illustrate this, I want to give a brief example, which is in the meerkat study system. So here you can see that the Kalahari Desert is actually a widely variable habitat. These are 3 different years: a wet year, a dry year, and an extremely dry year that happened in 2016-2017, where there was a terrible drought. And so how did this massive changes in environment structure affect the coordination system? So some colleagues of mine did a study on this at the time, and what they found was that due to the kind of decrease in the amount of resources available in the environment, what happened was that the meerkats tended to spread out farther apart from each other, searching for resources. And then what happened was that they adapted their communication system to try and, basically, we're thinking to try and stay together. So they increased their call rates, those cohesion calls that are kind of the social glue pulling them together. So this was an adaptation of a system behaviorally to a change in their environment. But did this actually work to keep the group together? As it turned out, not fully, because what ended up happening was that the rate of group splits, which are normally extremely rare in meerkats, increased by a factor of 3. So we think that potentially this was reflecting of an environment-induced failure of coordination in these systems, which might actually be important for the meerkats because, again, they rely on each other, especially for things like looking out for predators. So this is just like a small example, but I think it illustrates a general point that I think is very interesting and important for us to investigate in the future, which is that environmental change, of course, can affect individuals and in species, but we should also need to think about the ways in which environmental change might affect the communicational links between individuals and the ways in which they're socializing with one another. Because potentially when these communication links are affected, it could lead to aggregate changes in group structure, which can then have knock-on effects for survival and persistence. So moving forward, of course, the ways in which the environment is going to affect these coordination systems will depend a lot on the details of the coordination system. And so in order to understand these coordination processes, we really also need to take a cross-species approach to try and understand how different communication and coordination systems function. So we've already started doing this in our project. We are looking, as I mentioned in the beginning, at 2 other species, coatis here, shown on the left. Those are the kind of slightly less cohesive. And hyenas here, shown on the right. This is a much larger scale where they're mainly dispersed and they're kind of fissioning and fusioning with each other over time. We're really excited about these data sets. We only collected them a couple of months ago, so we're digging into them now. But I think looking forward, it would be really interesting to investigate these topics across a wider range of species in order to start asking more general questions about are there some general principles that we can think of that apply to coordination and communication systems across different species? And on the flip side, in what ways are these systems affected by their social and ecological constraints? And thinking also about the effects of environmental change, can we predict how flexible and robust these systems are going to be? Can we predict when these systems will be able to adapt to environmental change or when they might break down or change group structures? So with that, I would like to thank all of my collaborators on the Communication and Coordination Across Scales project, as well as my research group back in Konstanz, and thank you all for your attention.
Thank you, Ariana. Kind of reminded me of some of the studies that were done during the pandemic, if you remember, of cell phone tracing and how people were moving during the pandemic in areas that were restricted. So you are absolutely correct in terms of how people's movement are affected by the environment. So last talk in this segment, Cynthia Ismail on social network nodes as early signals for positive tipping points. Cynthia.
Okay, I hope my voice is clear. It's really thrilling experience for me to be here. A lot of chemical reactions in my body, just, to deliver my research findings. So, I just a little bit introduction here. My research is actually part of a European Horizon 2020 that we are a group of social scientists trying to understand a particular context, in this case coal and carbon intensive regions, and we want to provide empirical understanding how this context could shift towards sustainability. In this case, we use social ecological tipping points by adopting some of the fundamental theories from the natural science. This is just a slide, and I would assure you quite powerful that trigger where my research started. So starting from the left side, regarding SDG, I tried to picture now we have a really unprecedented challenge, which is climate change and sustainability issues. And this picture from Stockholm Resilience Center really helped me how to really break down what I have in order to really bring a solution to the, the body of knowledge. And then on the right side, this is, if I may take from the IPCC report, they try to conceptualize, the end goal of what we want in the future for our ecological, and now I want to add from our social system. Just a little bit of verbose slide, a conceptual background that we try to define positive social ecological tipping points from a social scientist. Here I want to cite my supervisor definition that those relatively small additional actions, in this case I will focus on the human nodes, or the policy intervention that at one moment could trigger an accelerates large systemic change. So in my case here, I still on the track to visualize the tipping points, but I will use certain elements here like the agency, human agency capacity, and their sense of direction, which is vision, as well as from the agent's capacity, I would like to see the intervention of the human. So it's a little bit different from the previous speaker. I will bring you to the human world. So this kind of like a background that at the end or in the beginning formulate my objective, and then I try to limit myself to just bring 2 questions. So the objective is try to better understanding like the interrelationship between visions and the social ecological networks as a signal for tipping points in the social system. And the research question is just to understand to which extent, this dynamic between visions or from the previous session narrative, also within the networks could really, trigger the transformation process in the social system, which is human, as part of the, Sustainable Development Goals. And the second research question is to how to track this kind of like really complex process. So I took Indonesia as a case study because first reason, this country is quite big. When you map out the global world, it's quite an eye-catching figure of this country. In terms of energy and electricity, it still consumes and delivers significant amount of coal. On the other hand, in terms of to tackle these unsustainability issues, this country already pledged to be net zero emission by 2060. So I perceive that this kind of contradictory perception implies we really need a radical change in human system. So just to be a little bit scientific, during the research, I deployed this framework by Weaver in 2006. And basically, this framework tried to guide me, like to bring a sustainability solution from your perspective. Basically, there are 3 stages, like from scoping, visioning, and then the experimenting. So during the first stage, which was quite challenging, especially during pandemic situation, I mean, you are social science and go out to observe. However, during pandemic, I cannot do the sightseeing or going out to do my research. And I had to be creative in this, like try to gather a lot of narrative and also information from all of the players within this energy sector, particularly electricity. So I see here webinar observation, because during that time, a lot of webinars. And I feel this strategy is quite useful as initial milestone for me before I conduct the set of strategy of interviews, survey, and FGD to know, who is really play a big role in shaping the transformation of the energy in the context. And also on the third stage, I did, gathering all of the different narratives and then translate it into discourse and put it into the position of networks, and you will see how the dynamics that could bring you the perspective of tipping element in social system. This is just a little bit elaboration of previous slide. So what I gained from doing the stakeholder engagement, webinar observation, and also in addition to desk research, I gather narrative as well as quantitative data. But here I will just focus more on the narrative because I want to show you, the main message of this presentation in this occasion. And from the narratives, I utilized a framework, like try to position narrative by the agents. And then, however, by understanding their position, it's not really, it only gives you a current situation, but we haven't understand the sense of direction for the future. So that's why I added Q methodology, if you are not understand, but we can talk more later on. But also I also introduce social ecological network analysis here, and I will explain more in the next slides. This is just a nice slides with people that I work with, trying to understand their narratives and translate it to understand this kind of process. So starting from 2020, a lot of interviews, workshops, and also FGD. We also work with policymakers in Indonesia, trying to give a two-way interaction, influencing one another. So these are just the people that I observe. And for this is just an example of how a form of Q methodology trying to code all of the narrative that I already gathered and try to code what kind of narrative or they really adopt in order to perceive the energy transformation. So the result of Q methodology here, I don't know if it's not working, but at least here among the human actors that I observe, at least there are two visions that drive the interaction among them in order to define what kind of energy transformation that they want to define as they want to be, as a positive one, or they do not want to be. Here, the first one, as the majority one, is they believe that tipping points, of course, clean energy should be there, but they still want the government as the leading. The other cluster of the group say that no, in fact, other kind of agents still have their own capacity in order to trigger the change, like private sector and civil society. And then here, I don't know why the slide is a little bit, it's blank, maybe can be used. But here in the poster, unfortunately, somehow it's gone. But the networks, I can't see that there are at least, okay, there you are, thank you very much. So on the left side here, using a narrative positioning, and I position the agents. So basically, we have quite a diverse agent with their own narrative. And in terms of transformation, they have their own position in that. And I just want to pinpoint, particularly for government, I would say they are still in the dilemma, whether still keep the coal or moving towards the clean energy. However, there are still other agents like NGOs or local companies that's already quite brave to really raise their voice. So let's move to the clean energy. And then when I utilize the network theory and deploy centrality in the networks, you can see here two human actors or institution is quite the central of how this energy works in the context. So for the first one, I introduced you, PLN here is the state-owned electricity company, but sometimes can act as a government. The other one is Ministry of Energy and Mineral Resources. So if you remember the first vision, so it's really obvious how the human actors here really make an interaction one to another based on what vision they want. So because many of them perceive that government is really a key role here. So you can see here the Ministry of Energy and the state-owned electricity company is quite a big role. However, if we look at the position of their narrative, they are still in dilemma. So that's why one of the participants said, we keep trying to make interaction with them, even though currently they still have not replied with, like open way communication. So it's quite interesting. And then I just want to highlight here, I added the element of region as the limit of, how the human can behave, because obviously the ecology is our boundary. And it turns out that the agents, like Ministry of Energy here, doesn't really have a direct control to the particularly a region here, the Banten province, that I'm focusing mainly. And then, in fact, there are other agents like civil society and local companies that they really raise their voice that I don't really want coal power plants in the back of my yard, or even geothermal because of the historical failure of geothermal power plants in my back of yard. So if we add the element of region here, it's quite clear that some agents do not really have control of how the transformation could happen in certain contexts. So you can see here how I just want to extend the message from the previous session that how narrative could be really powerful in order to to make a transformation happen towards sustainability. And in fact, I want to promote that networks here, by understanding how networks here in our society, we could know like maybe tipping elements could happen by just switch or just swap the narrative of a certain agents, and they could do your visions because they have certain capacity that you might not have. So it's quite interesting how I observe, the work of this research. And then just to give you an insight, so actually the transformative do not only have one single particular vision. There are many and complex problems like climate change. We have to use all of mixed method approach. And there are no one single solution for this complex challenge. And if I may add, do not underestimate the weak ties agents like civil society or local companies. And then if I may add another thing is, if you notice from the previous slides that from IPCC report, my research, if I could add a little bit, IPCC report already add a vision, like our end goal, what we want to be. And in fact, some of scientists use vision, what they want to be in their context of research. And I just want to add that no single vision in order to bring us to the end of our goal. So this is my addition for my research. And as a thank you, I just want to head up for my supervisors over there, and also the consortium members that really give me opportunity to explore and contribute in this research area. And last but not least, just to extend the message of one of the speakers during the first day, she said that just give the researchers money to do something. But as social scientists, for me, just give some of your time, so let's see what we can do together to change. Thank you very much.
Thank you very much. So I think you've been hearing across the entire program the progression from what can we do, how do we communicate, and how do we turn communication into action. And that's what you're also seeing in this session, We heard last night a call to action by our dancers who showed that movement and rhythm can actually induce communication and asked, how does our brain do that? And you've heard already something about the development of the systems where that happens, and then an observation of those kinds of behaviors in the wild, and now a movement from those forms of communication into tipping points. And we'll continue now with our next keynote, which is going to be given by Ulf Diekmann of the Okinawa Institute of Science and Technology in Japan and the International Institute for Applied Systems Analysis in Luxembourg, Austria. And he's going to tell us about a systems perspective on human behavior beyond discipline boundaries. Thank you.
Thank you very much, Marina, for your kind introduction. I would like to take as my point of departure the 4 claims or theses or tenets I presented you with on the first day of this event, the summit. And we all take now as the agenda for sustainable development and environmental protection and responsible conduct in the biosphere, the SDGs, the Sustainable Development Goals. And when you analyze these, you soon find out that essentially all of them, maybe most of them, are connected to social dilemmas which result when selfish actors jeopardize common goods, according to the paradigm of the tragedy of the commons. And in order to mitigate such situations, we need quantitative models, because essentially our linearly thinking and intuition geared brains are not quite suitable for understanding all the complexities and all the feedbacks that are involved in the complex systems we have to manage. And in devising these models, we have big challenges, because while stylized models are already working well, we need to incorporate behavioral, social, and institutional dynamics into these models, and we need to do that with realism that enables predictability. And as I said on the first day of the summit, this requires a further unification of the behavioral science across all these disciplines and fields listed here. So in particular, I will mention again in a few slides evolution, economics, and cognitive science. And with that, I switch to the second part of my introduction, which is about system thinking. You saw the term systems perspectives in the title of my talk. So what is system thinking? I think we all have a notion and some intuition what it means. So let me just run over a few basics so that we are all on the same page. The main picture that you need to appreciate in a systems context are connected subsystems in interactions and errors and boundaries. So I've put this into this little table for illustration. We can look at all the levels of our sustainability challenges, from the very global level of the biosphere to the national level of federal decision-taking, to regional levels, communities, and all the way down to individual levels like households. The actors differ, the agency differs. We have supranatural institutions, then we have governments, NGOs, country officials, we have regional private entities, but also regional public entities, and then, as mentioned, households, firms, and banks. And often they interact in complex ways, horizontally within one level and vertically across levels. So this system of systems framing is often really helpful to make sure that essential parts of the analysis are not left out. And further on, to gear our minds in the right way, we want to be inclusive in this analysis. And here's a kind of mental laundry list you can use when you are faced with a complex problem, and just quickly go over these different dimensions and make sure you cover as much as possible, as much as needed from them. So this takes us from the impacts. Usually there's not just one impact, for example, on the biodiversity protection, but also related impacts on, say, food provisioning. Then feedbacks are central. Arise from the microscopic interactions in complex systems and often have a surprising character, so we must take them into account as much as possible in our foresight. And then all of these systems have stakeholders. There's not just one convenient social planner that needs to be satisfied, but different stakeholders with different values, with different beliefs and different interests that we need to take into account. And then carrying on, we can further maybe broaden the horizon as needed. We have to think about sectors. We might think of the energy sector, but it's related to the water provision, it's related to agriculture, so going over that kind of nexus is really helpful to establish a good systems perspectives. Then governance dimensions, you might have to deal with a local authority, but then there are regulations from a national authority to keep in mind, so that is also useful. Disciplinary and interdisciplinary perspectives have to be combined, no surprise there. And ultimately, you want to transcend a single scale and connect the systems analysis across multiple scales in the vertical direction I showed before. Furthermore, systems analysis is open and methodologically open and pluralistic. So it's useful to think in qualitative terms, but that usually needs to be combined with quantitative methods. It's also useful to think in terms of stylized models, which often bring out certain phenomena very clearly, but often ultimately then for prediction and management, we need complex models. And all of this has to be brought together in a coherent framework, which is often based on narratives and case studies. Now, the systems perspective, of course, is not new. It has been recognized in all the disciplines in different ways, under different terms, and I start here with one of the earliest in this regard, the scissor metaphor of Herbert Simon, who is also the father of the bounded rationality concept. And he cogently observed that if we want to understand human cognition and human behavior, it's always in the context of environmental conditions and constraints that we operate, that our mind is formed, that our decisions are taken. So behavior is the juncture of the cognition with the environment, and neither blade of the scissor can cut alone. They have to be considered together. A little bit later, the field of cognitive science recognized the importance of embodied intelligence. How intelligent, how capable can an artificial intelligence be that is just trained without having a body, without having interactions with the environment, without the ability to probe and experience? So feedback interactions with the environment are recognized as central for building an intelligence. And when you look at these encoder-decoder models or general language models, there's a real debate how far they can go without having such embodiment. In the context of evolutionary theory, there's a framework of adaptive dynamics which comes forward with a similar clear pronouncement that we need to consider resident biological agents in ecology as modifying their environment. The term here is niche construction or frequency dependence. And then this environment is experienced by variants, genetic variants, sometimes phenotypic variants. And if they are successful in the environment set by the residents, then they can invade and sometimes replace the residents. This is what we call eco-evolutionary feedback. So all of these are examples about the interaction between subsystems in the context of a broader environment. And with this, I would like to move to the main part of my talk. I've selected 5 vignettes that illustrate, from my personal perspective, the current research challenges for building realistic models of human cooperation. This is not an exhaustive list. I think there are quite a few more challenges, but just to give you an impression, I think these are some of the most important ones. Let me start from the idea that strategies, decisions we take, are usually in a continuous context. They are graded. You have to decide how much time you invest in a task. You have to decide how much money you give to a certain entity. You have to decide how far you go to maybe buy a product. So it's not on and off decisions. It's not binary. And unfortunately, almost all of classical game theory that is still a very active field today is built on stylized strategies that are binary. So famously, you have cooperators and defectors. And that gains a lot of knowledge and important knowledge, but there's more to human interactions than binary strategies, and that also takes us to the field of social norms, which are usually graded. Then there are so many mechanisms that can promote cooperation. It's actually an embarrassment of riches. You can change the structure of a population, the assortment of interactions. You can put regulations on the agents. You can give positive incentives, you can give negative incentives. You can allow agents to have a voluntary contribution or a mandatory contribution. And over the last 10, 20 years, these aspects have been investigated. More and more comes to the fore, but there's still a big gap in bringing them all together in realistic models that are not just focusing on one mechanism, but on the entanglement of these mechanisms in realistic social dynamics. Then there is multi-level selection that takes us back to the system perspective and the vertical dimension. We have agents that are reporting or regulated by agencies, and these agencies or institutions are often there to ensure a cooperative functioning of the system. But in such a context, you also have to wonder, how does the institution keep functioning? Who is guarding the guardians? Who is making sure that the institutions are doing what they should do? Then we come to the big field of bounded rationality. Bounded rationality is a polite term for various degrees of irrationality. We saw quite a lot of that in the context of the COVID-19 crisis and the public response to that. So certainly we are by no means optimizing agents that have some sort of utility functions coded into our minds and act according to this paradigm of the homo economicus, or the rational actor model. And then there's plural rationality. I said we don't have a single social planner. Usually we have multiple stakeholders, and addressing this stakeholder diversity is imperative for cooperation models that have any bearing on reality. So I now walk you through these 5 particulars and start with continuous strategies and evolving social norms. So what is a social norm? A social norm often translates an observed feature in your environment to action, to a decision. And in the context of cooperation models, the 2 main features that are to be considered, and which I link here in this picture, is the level of cooperation that you observe in other agents. How much do they give to the common good? How much, for example, do they help in cleaning the dishes from a communal kitchen, or how much do they help with mitigating climate change by reducing emissions? And then you have an incentive on the vertical axis, which can be positive in form of rewarding or negative in the form of punishment. Here, the illustration is on punishment. Now you need to link the 2. So at a given level of cooperation or acting according to expectations and rules, what is your institutional, what is your sanctioning response? And when you link this by the sigmoid curve you can see in this slide, then you realize this is actually an emergent definition of defectors and cooperators. So the defectors are those, by definition of this curve, that are giving too little and therefore are sanctioned, and the cooperators are those who give enough and therefore are free from this negative incentive. So that takes us very quickly to the perspective of letting this social norm evolve. And in practical terms, you can do that by 4 scalar quantities. We have the cooperation level, that's the primary trait, you want to bring that up. And then we have 3 traits or features or strategy components which determine the inflection point of this sigmoid curve. So where do you cease to punish? The severity of the punishment, that is the maximum of the curve, and the strictness of the punishment, which is the steepness of the curve at the inflection point. Now we can put that into an individual-based or agent-based model, crank the handle with a public goods game as the basis, and see what happens. And it's quite remarkable. You can start from a society of agents that do not cooperate and do not punish. That is a completely ancestral social void, if you will. One could imagine a tribal society maybe starting out from such a setting. And then there's bootstrapping. Bootstrapping means that starting from zero, you get going with cooperation through punishment. Actually, many models of cooperation cannot do that, so it's quite a feature, because in many models of cooperation, you need to exceed a certain threshold of cooperativeness before cooperativeness takes on further. So here you can start from literally zero and gear it all up to a rise of cooperation through the thread of punishment, which keeps pushing cooperators up. And that's also very interesting. It means that the evolution of the social norm implies that early in the game, a certain investment may be enough, and you are qualified as a cooperator who doesn't get punished. But late in the game, expectations have risen. The group has moved on to higher standards of cooperativeness, and you need to invest more in order not to get punished. This is actually related to the idea of keeping certain international environmental agreements dynamic and making the expectations contingent on the level of compliance. I think there is a lot of potential in this idea. The second example is about the interaction between different mechanisms and harnessing their synergies. So here you have three fundamental incentive policies that one could consider when you are confronted with a cooperative challenge. And for the sake of completeness, maybe let's consider a classroom which is very unruly and you want to bring it to order. And you could start by rewarding individual students. All of them are kind of shouting and running, and you start to reward individual students who are behaving well. That is good for getting the cooperation off the ground. So this jump starting of cooperation through rewarding works very well. But then to keep cooperation rising up and eventually to keep it going to 100% through rewarding is very difficult. You keep rewarding everybody, it kind of loses its differential ability, and it's also very costly. Now, you could think about punishing. So punishing is very good when we are close to full cooperation. You just pick out the few unruly agents and invest your institutional budget or your patience in negative incentives, and you bring them back to order. Works very well. But if you start from an uncooperative state of the system, this is not working at all. There's a threshold, like the one I mentioned before, of cooperativeness you need to exceed before punishment takes you in the right direction. Otherwise, it actually takes you in the wrong direction. So now it's quite an obvious idea, but a few years ago when we did it, it was the first time it was done to combine these two and to try to harness the advantages of the one without losing the advantage of the other. Trying to go back because it went forward without my doing. There we go back. So this is what we call, to kind of to coin a catchy phrase, first carrot, then stick. You know the carrot and stick approach. It's a bit more specific than that. It's first carrot, then stick. So you start with rewarding, and you then, at a certain point, switch to punishing. And you see in this diagram here, it has terrific properties when you do that. You can jumpstart cooperation. So you get the cooperation off the ground easily with rewarding. At a certain point, you switch to punishing and you bring it to completion. And when you look at the horizontal axis, the incentive strength, you need far less money or resources or patience to do that with this hybrid strategy than with the rewarding strategy, and certainly than with the punishing strategy. So that seems like a good idea, and it's actually not so frequently used in practice, in institutional practice. We can corroborate that this is a very good idea by looking at the effectiveness and efficiency. Effectiveness means under how many different situations does it work. These are spread out in this square here, and if we can bring the frequency of cooperators, which is used as an indicator on the left-hand side, high up to green, that's good. So we want to have a large green area, and you see the green area is largest for the first carrot then stick approach. And then we also want to be efficient, so the cumulative cost of the enterprise should not be too high. Again, green is good, and you see the first carrot then stick is far better than either rewarding alone or punishing alone. We can also put this into a spatial context. In reality, we would probably have to think about social networks rather than grids, but the principles are the same. If you have rewarding alone, then you get these clusters, these diluted clusters of cooperators. that eventually cover the whole system but never go to full cooperation. Punishing alone produces compact clusters of cooperators which do go to 100% cooperation but only work with high incentives. And when you combine the two, you switch from rewarding first to punishing later, then you get the best of both worlds. You get first these diluted clusters which create the conditions for the compact clusters of cooperators to emerge in a kind of booster stage of the process. I now come to the third example about multi-level selection and what we can do to keep institutions functioning. This is research in its infancy. It's a model we published in PNAS just a couple of years ago, which was motivated by recognizing that much of evolutionary game theory assumes that these incentives I have now been talking about, once established, get implemented correctly, or only with random errors. So you have incentives, they help cooperation, but how about the reliability of the incentives? Maybe something goes wrong with the incentivizing agents or the incentivizing institution. So in real life, incentives provided by institutions are subject to the risk of corruption, unfortunately. A lot of developmental aid is not as efficient as it could because of such issues. That potentially jeopardizes the pro-social effects of these incentives for obvious reasons. So what influences the dynamics of institutional corruption? Can it be avoided? And if so, how? Very big questions which we cannot answer, but we built a first game theoretical model of the problem. Again, one would wonder, such a big problem, obvious relevance, but nobody had done it before, so we did this little bit of pioneering work, and it's very simple and stylized, but it does give insights. So we have different types of cooperators and defectors, which are either looking at the reputation of the umpires, which is the institution, or not. So being cognizant of how honest or corrupt the umpires or the institution are is a key mechanism in this model, and we assume that agents can invest a certain amount of money to find out about this state of the institution. So if they pay this money, they can learn that their umpire might not be honest, and therefore maybe they don't engage the service of the umpire to mitigate their cooperative gains. That's the basic system here. And you see these tetrahedra. These are the simplices for 4 discrete strategies. So unfortunately here we are back to discrete strategies. And in each tetrahedron you have either honest umpires, fully honest umpires, or fully corrupt umpires, left and right. And these are connected by horizontal lines which describe the frequency of corrupt umpires going from left to right. So then you can see that under certain conditions you have damped oscillations to a fixed point with actually rather high levels of corruption, or you can have cycles, limit cycles, in this simplex of the game that go through the prudent cooperators, which are scrutinizing the integrity of the institution. First to optimistic cooperators, which are foregoing this scrutiny in order to save money, because they think it works anyway. Yeah? So in a system where corruption is low, investing into the scrutiny is not necessary. But that's the road to doom, because once you have the loss of the scrutiny, corruption shoots up. That's the upper part of the orange curve. That brings back the prudency, the scrutiny of the institution, and that brings then back the honesty of the institution. So we see that, I think, in many real-life contexts, maybe not as extreme as in this stylized model, but when things are going well, it's always hard for politicians to argue to keep investing into scrutiny. Military investments come to mind, which now recently have taken direction towards increased scrutiny, increased vigilance for the needs of that becoming more obvious again. I come to the fourth example, and that is about bounded rationality. Again, a very big topic, going beyond the homo economicus or rational actor model assumption of much of stylized theory. So the example here is based on modeling systemic risk. And you might recall from the global economic crisis of 2008-2009 that systemic risk took a spotlight then when banks defaulted and spread their defaults to other banks to which they were economically connected. That is a paradigm of cascading failures, which implies systemic risk. It's very amenable to theoretical studies using network theory and agent-based models. So here we have a little network. In practice, of course, we study much larger networks, and we have one failure. And then this failure can spread to another connected agent, let's say a bank that has economic ties to the failed bank. And then with a certain probability, in this simple case 50%, this then also fails. And then this can spread further across all links, and with a certain probability failure occurs until eventually the cascade saturates and we have a certain number of failed nodes. Now this is really important for many practical applications in complex systems because they are highly connected and there are cascading failures that can occur. So how good are we in understanding this? Well, it turns out not so very good, and maybe you're not surprised to hear that. It's not an easy challenge. So we assembled a web-based survey in which we showed people such a network. You see it here on the left. And then we asked them certain questions about the networks. We collected more than 2,500 answers and analyzed these. So here are the questions we ask. Let's look at one of them. When node X fails, what is the % probability that node Y fails? Well, that's the most basic question of systemic risk analysis. And if you are confronted with seeing this network, it's really not easy to come up with a judgment. You need some intuition, and our intuition is not terribly good. The second question is even more pertinent when one random node fails, because you might not have enough knowledge to pinpoint the most likely failure. So if one random node fails, what is the % probability that you are affected or that node X fails? And then there are other questions. So the result of this survey is that in general, systemic risk is quite terribly underestimated, very systematically underestimated by about 38%. We can translate that into a log odds ratio of minus 0.5 roughly. So that's the overall picture. We are all not good, but it's not that we randomly do wrong, we very systematically do wrong in the direction of underestimating risk. And I think this speaks to a lot of economic developments we see. And then if you compare the numbers for the different questions, it's only 0.3 for the simple question, but the 0.7, so something like 80% underestimation for the more difficult question when a random node fails. So not an easy question. This varies with the type of network we present, high and low connectivity. So with higher connectivity, it's even harder. So by increasing the number of connections by 50%, actually the underestimation shoot up by 400%. So it's quite dramatic. More connections mean that we underestimate even more severely. And then in these questions, when we name particular nodes, the underestimation is a bit better, so less underestimation, but when we ask for random nodes, it becomes even worse. Surprisingly, we even found effects of gender, of mood, which we did collect at the beginning, and of age. So all this shows that systemic risk, being a very important property, is not easily estimated by humans. And with that, I come to the fifth example, plural rationality, addressing stakeholder diversity. So here I take the context of fisheries management, and you know the yield curve translates the fishing effort you put into certain benefits. And the yellow curve is famous, the yield curve. If you do not fish, you have no yield. If you fish too much, you cause the population to crash, and in between you have a maximum. The Gordon-Scheffer model of resource management. Then profit peaks to the left of yield because you have costs. Employment goes up all the way because more effort requires more work, and ecosystem preservation goes down. So if you look at these four key indicators of utility, then it turns out that fisheries management has traditionally operated on the right-hand side and now is gradually, slowly shifting to the left-hand side where rational analysis actually indicates that it should be. So to put this into practice, because this was just a qualitative statement by Ray Hillman, we built a model that combined the biological, the socioeconomic, and the stakeholder complexity. You see the biological model feeding into the socio-ecological model. That gives utility components of the 4 types are indicated, and then we view these utility components from the perspective of different stakeholder groups, construct stakeholder-specific total utilities, and then aggregate those further into what we call joint stakeholder satisfaction. In order to base this on realistic preferences, we extracted from the literature and from newspapers estimates of how much different stakeholders care about these utility components and collected them in this matrix. And then cranking the handle, we can plot the joint stakeholder satisfaction as a function of 2 control variables that managers can tune. The harvest proportion, or total allowable catches on the horizontal axis, and the minimum size limit, which regulates the gear that you are allowed to use as a fisherman, how large the mesh size must be. You see in color, in green, the good parts of the control space where the joint stakeholder satisfaction is high, and in red the bad ones, and in gray the unfeasible or collapsed ones. The stars or asterisks indicate where we predict the joint stakeholder satisfaction to be highest, and the circles are where the systems are currently operating. So you see quite a gap here. So taking these stakeholder satisfaction quantitative assessments into account should enable us to give better advice and do better resource management. And with that, I come to my last slide and the summary. I hope I have illustrated that models of human cooperation can serve as integrative, cross-disciplinary devices for exploring solutions to global challenges. A fruitful interface between stylized mathematical models and complex integrative assessment models is gradually developing. And much work remains to be done on understanding and incorporating sufficiently realistic behavioral, social, and institutional dynamics. Thank you very much for your attention.
Thank you very much. So we've heard about how we can look at systems and try to push the parameters of systems into appropriate use of resources. And now we'll hear about disparities. And particularly, we will hear about the dances of the Caribbean, cancer disparities in people of African ancestry. And that will be given by Juliet Daniel, who's from McMaster University in Canada.
Okay, good afternoon, everyone, and thank you all for hanging in there and sticking it out to the end. I'd like to just thank the organizers for inviting me to speak today, and those of you that are probably wondering what does cancer and disparities have to do with climate change and all these other topics we've been talking about. I've just put some of the UN, the Sustainable Development Goals that this particular project I'm working on, I think, is most relevant to. So I wanted to start by just giving a little story about myself and how I got to be doing what I'm doing. I was born and raised in the beautiful island of Barbados in the Caribbean. I came from a low-income family, but my parents and my neighbors and everyone recognized I was a unicorn and quite different. And so they saved as much as they could to send me to university for the first year. And I chose Queen's University in Canada because they acknowledged me as an individual before I even got there, because this was pre-internet, Google, everything, and we couldn't afford to go see the universities. But they were the only university I applied to that actually sent me information every other week before I had to make a final decision as to which university to attend. So I went to Queen's, had a marvelous time. However, in the last year of my, in the last year of my undergrad degree, my neighbor in Barbados passed away from breast cancer. My mother was diagnosed with ovarian cancer, and then she succumbed to ovarian cancer 6 months later and 5 days before I convocated from Queen's. So at that point, I was heading towards medical school, and I decided to pursue cancer research instead. because I felt that the doctors were not able to save her because we did not have enough drugs and therapies for ovarian cancer. And sadly, we still don't have enough drugs or therapies for aggressive cancers like ovarian cancer or pancreatic cancer. At that point, the novelty of Canada and the cold weather had worn off, and I chose to do a postdoctoral fellowship in Memphis, Tennessee, because I thought it was a lot further south and would be warmer, like the Caribbean. And I chose to work in Dr. Al Reynolds' lab because during my graduate studies at University of British Columbia, I realized that the one hallmark of cancer that really led to the demise of most patients was the fact that the tumors broke away from their primary tumor site and spread to other organs such as the lungs, the liver, and the brain. So Dr. Reynolds at the time had just discovered a new gene himself, and it was one of the first substrates that was identified for one of the first oncogenes that was identified in the mid-80s that contributed to human cancers. But no one knew how the SARC kinase actually caused cancer. And Al was part of the team in Virginia that identified many of the SARC substrates that people around the world are now studying. And so I chose to work in Al's lab because he was the only person studying P120, and I felt that anything I did in his lab would actually have some impact. So he was studying this protein. Where's the pointer? Is this? Yeah. So he was studying this protein P120 that he identified as associating with a transmembrane cell adhesion protein, E-cadherin, and 50% of all human metastatic tumors are due to malfunction of that E-cadherin-catenin complex. So my role in his lab, or my project, was to determine the role of this new protein that he had identified to understand how it was causing metastasis in human tumors. So during the process of characterizing P120, I cloned a novel gene, just like he did, that was not characterized. And he gave me permission to name it anything I wanted. Being from the Caribbean, not seeing many black women, far less black scientists, I decided to name it something that would be a little, Caribbean. And so I named it Kaiso after my favorite calypso music. And this is just a photo of the dancers in the Caribbean and in Toronto when we have our annual Caribbean Festival. And over the first 8 years or so as a faculty, why are the slides all kind of weird? Okay. It's not going back. Okay, we'll just leave it there. Okay, so the first few years as an independent professor, I spent my time characterizing Kaiso, determining what it did, and determined that it was a dual specificity transcription factor, meaning that unlike most transcription factors, it bound to 2 different types of DNA, which meant that it regulated many more genes than a typical transcription factor did, which meant it would also be harder to really characterize in depth, but it meant that it was not a low-hanging fruit. We also found that it was implicated in many biological processes. Most of these were all processes that contributed to cancer: cell proliferation, cell motility, inflammation, etc. And my lab and others found that it played a role in multiple cancers. So while I was doing this, I kept thinking, this is not why I pursued a PhD. I really wanted to do research that would have global impact. That was my childhood dream growing up in Barbados. And I felt like I was doing fundamental research, which was great, but I wasn't seeing that human application. And then in 2008, I attended the American Association for Cancer Research Conference and heard the physician there, Dr. Lisa Newman, talk about a breast cancer subtype that was disproportionately affecting Black women in America as well as in West Africa. And that really piqued my interest because I had never heard about this, even though I'd been doing cancer research for over a decade. And at McMaster, they were also encouraging us to think about retirement/refreshment. So I was already thinking about what can I do to give back to the Caribbean with my cancer research efforts. Oh, the slides just they were fine earlier. Anyway, so breast cancers are classified based on the expression of 3 biomarkers that some of you, hopefully, or all of you have heard about: the estrogen receptor, the progesterone receptor, and the HER2 receptor, which should have been here, but you can't see them. But basically, this is the cell. These are the 3 receptors. And the subtype of breast cancer, triple-negative breast cancer, those cancers or tumors do not express any of those biomarkers. And the reason that this is problematic is because the current treatments for breast cancer specifically target estrogen receptor or the HER2 receptor. So we have tamoxifen that targets estrogen receptor or ER, and Herceptin that targets for the HER2. Triple-negative breast cancer patients, however, do not express any of these biomarkers, consequently cannot be treated with these drugs, and can only be treated with standard chemotherapy or radiation therapy. And as a consequence of this, they have the highest mortality rate of all diagnosed breast cancers. As I said, when I first heard about this, heard Lisa Newman talk about this was really disturbing, one, being a Black woman, and coming from the Caribbean, where I know that we were ancestrally linked to women in West Africa, where she was seeing an incidence of triple-negative breast cancer as high as 50% in the women that were going to the hospitals there. So what is triple-negative breast cancer? I've just told you. So it's highly prevalent in young premenopausal Black women, as well as some young premenopausal Hispanic women. There's a poor prognosis, which we know is due to the lack of these specific therapies because they don't express ER or HER2. And initially, when I first heard Dr. Newman talk about this and when I read the literature, most of the studies were suggesting or implying that this poor mortality and high prevalence was due simply to the low socioeconomic status of Black women in West Africa and in the United States. So while socioeconomic status might explain the outcomes and poor mortality or high mortality rates, it wouldn't necessarily explain the high incidence in these populations. Notably, the World Health Organization has indicated that breast cancer is actually an emerging health crisis in Africa, since the 5-year survival from breast cancer in Africa, in sub-Saharan Africa, is less than 50%, compared to 90% in North America and European countries. So this is really problematic. Sadly, the US is one of the only places that collects race-based data. So most of our studies and the studies of those of us studying triple-negative breast cancer are based on data collected in the US, because in Canada, for example, we do not collect race-based data. We've just started to collect race-based data, and it will take at least 5 to 10 years before we have enough data to analyze in the Canadian context. But I wanted to show this slide to show you that, historically, NHW refers to non-Hispanic white or white women, and NHB is non-Hispanic Black women. So historically, white women have had the highest incidence of breast cancer since they started collecting the data. This graph doesn't show it, but they've been collecting this data since 1975 in the US. And it has always been predominantly white women with the highest incidence of breast cancer. And hopefully, and on this graph here, which is now showing the trends in mortality, you can see that there's suddenly this switch from white women being at the top of the graph. You see this sudden decline in the mortality rates for white women, but the mortality rates for Black women kept increasing slightly and then started to decrease, but they remained the highest mortality rate of all the ethnic groups in the US and significantly higher than all of these other groups. We believe, or I think we're pretty confident, that this decline that we see here in white women was due to the introduction and the development of tamoxifen and Herceptin, which most white women tend to express those biomarkers. They tend to have ER-positive tumors or HER2-positive tumors. And sadly, most of the clinical trials that were done when these drugs were being developed were done predominantly on white women and not diverse populations. And because if they had been, they might have noticed that the Black women were not responding to those drugs. Next slide. So as I said, I first heard Dr. Newman in 2008. My next research leave was scheduled for 2011. So in 2011, I went to Barbados and started collaborating with physicians there, as well as in Nigeria. And this is just a diagram of a schematic of the globe showing you the incidence of triple-negative breast cancer based in, West Africa? Yep. So in West Africa, the incidence of triple-negative ranges from 50 to 60%. This 15% is predominantly in East Africa, which is not where most of the transatlantic slaves originated from. So we believe that the high incidence in the Caribbean and in Black American women is due to being descendants of these ancestors in West Africa. So as I said, I started collaborating with oncologists and researchers and pathologists in the Caribbean as well as in Nigeria. And we collected triple-negative breast cancer samples from these women. And then we did this high-throughput study called tissue microarray analysis, where we put small pieces of tissues from each of these patients onto one slide, which allowed us to analyze the expression of different genes and biomarkers simultaneously on multiple patients, rather than doing it one at a time, which was historically how it was done in the past. And in the interest of time, I just wanted to show you the results that we found when we looked at the expression of the gene Kaizo that I had cloned and discovered. And the reason we looked at this was I had collaborated with a scientist in the Netherlands, and we had found that Dutch women with very aggressive invasive ductal carcinomas had significantly higher levels of Kaizo expression compared to Dutch women with non-invasive type cancers. And in this, I hope you can appreciate and see in this slide that the Nigerian and Barbadian women had significantly more Kaizo expression, as indicated by the dark brown staining in the nucleus and the cytoplasm of the cells, compared to African American women and compared to Caucasian women. Oh goodness, I can't believe these slides they were fine. Anyway, so alongside while we were doing this, we also looked at the clinical pathological variables of women in Barbados diagnosed with triple-negative breast cancer. And here, unfortunately, you can't see it. My gosh. Am I supposed to be pointing at this, perhaps? Maybe. But the red rectangle there was highlighting the fact that triple-negative breast cancer patients in Barbados had the highest grade tumors. As I said, because we were noticing this high level of Kaizo expression, we then generated some Kaizo-depleted cell lines to see whether that would cause morphological changes in the cells, which they did. And you can see here, these are the parental, highly metastatic cells expressing lots of Kaizo, very little of that protein E-cadherin that mediates cell-cell adhesion. When we deplete or remove Kaizo, we get the restoration of E-cadherin, and the cells become epithelial again, which means they're less motile. We then confirmed this using mouse models where we put these Kaizo-depleted as well as the parental cells into nude mice, into the breast mammary fat pads of the mice, and then looked to see whether the removal of Kaizo would prevent the metastasis or spread of these triple-negative breast cancer cells to the lungs and the liver. And we were pleasantly surprised and excited to find that indeed the simple removal of Kaizo prevented the metastasis of these tumors. As you can see here, the parental cells had lots of metastasis in the lungs and the liver, indicated by these darker pink regions outlined in white, whereas the mice that had the Kaizo-depleted triple-negative cells hardly had any metastasis. So these are just some of our ongoing and future studies. We're trying to determine the cause of high Kaizo expression in aggressive breast cancers. We're developing prognostic and diagnostic kits to see if we can look at Kaizo expression to help physicians and women determine whether or not the types of therapies and perhaps surgeries that they should consider. And we're characterizing some of the genomic profiles of women in the Caribbean. And just to wrap up, I just wanted to mention, in case you didn't realize or know, that we're wrapping up the end of the International Decade for People of African Ancestry or Descent, which represents about 1.5 billion people globally and 18% of the global population. And again, because most of this work is being done in the US, this is a statement from the American Association for Cancer Research in their annual Cancer Disparities report. This is from 2020, where they've added racism trauma as a social determinant of health. And they've added this to determinants such as education, income, etc. So a few years ago, in the theme of the Human Frontiers, Canada has adopted this approach. And we now have a New Frontiers program, funding program in Canada, and I was fortunate enough to be funded for this program along with two collaborators, Dr. Ingrid Waldron, who is a social scientist, and Paula Marignani, who is another molecular biologist like myself. And we're doing this in partnership with a community member from this small community in Nova Scotia called Shelburne. And the Shelburne community is a community that was established 400 plus years ago by slaves that were brought over from West Africa, as well as some were brought from the Caribbean. And this small community has an incredibly high and unusually high incidence of multiple different types of cancers. And they believe that it could be due to toxic chemicals that are in their community, since a dump, unfortunately a biohazard dump, was put in that community 75 years ago. And so we're doing the study to try to see whether or not that is a contributing factor to the high incidence of cancer, or whether it's due to perhaps some ancestral mutations that they have. And this is just some recommendations in general. If we want to improve the health of the planet, we need to definitely address and improve Black women's health in particular, because we're suffering, and we want to contribute to society. And so we need increased representation of Black healthcare professionals and researchers. There's not enough of us, definitely not in Canada. And I think it's just me and some of my trainees. And just to say that if we do this and we have some representation with agency, I heard everyone talking about more inclusive stakeholders and diversity. And so I'm just, as I said, I'm just trying to showcase that if we can tackle this problem together, we will have this beautiful world, hopefully, that we can continue to live on for longer than we anticipate at the moment. And I'll end there. Thank you. Oops. I'll put it there. Yes. Oh, yeah. Forgot.
Please sit. So please, can we have the panelists back up on the stage? Now we have time for discussion. And I think that the concluding remarks that you heard were especially apt as we think about the disproportionate effects of climate change on populations that are already struggling, whether these are ethnic minorities in the developed world, whether this is sub-Saharan Africa or South America, where the effects of climate are going to be incredibly profound much more quickly. And I think that what you've heard is about how all of the science that we've been discussing around the rapidity of change, on the mitigation of change, really depends on human actors and on understanding the challenges to human communication and human action. So I will turn it over to my colleague to start off our panel.
Sure. Thank you, Marina. And I've been fortunate to be able to talk with most of you at points throughout the conference, and you've seen in the last hour or so, hour and a half, this has been quite a journey in terms of the research that's being done and how it's being done. But I also know, in speaking with these amazing individuals, they've also taken quite a journey in terms of where they ended up disciplinarily and in terms of the subject matter that they're focusing on, which is quite sophisticated, as you've seen. So my first question for them is really about the kinds of issues you've faced in working across disciplines. Because some of the stuff you're doing, it's pretty groundbreaking. The New Frontiers program, second plug, was designed specifically to get at this sort of broader reach, interdisciplinarity, transdisciplinarity. That's what it was for. That's what it was designed for, even though only about 10 or 15% of projects that are funded under our New Frontiers project really get to that point. But that's what it was supposed to do, and I think these are brilliant examples, even though we've only funded one of you, of the kind of work that we would fund. Oh, and also, you can be a co-applicant and be funded as long as you have a Canadian partner. So, okay, I'm done. What kind of issues, have you faced in getting where you got?
Do you want to go this way? Okay, sure. So I think I would say, so first of all, when I first started the project on triple-negative breast cancer, when I first applied for funding, the very first year, it was rejected, and the reviewer said that it wasn't relevant to the Canadian context. And that was incredibly devastating because, one, we had Black women in Canada, and secondly, as I said, we've got over, probably close to 1 billion Black women globally. But I don't give up, so I kept trying, and I got funded third time. And then I would say for the New Frontiers project, as I said, we were funded 2 years ago, and the challenge has been getting ethics approval. So although the application specifically said the project was triggered by the community reaching out to the social scientist, who then reached out to a geneticist and molecular biologists like me, the ethics board was upset that there was a community partner who had the list of names of people that had cancer. And so that it literally took us a year to get ethics. And so we're now starting that project because we had to explain, if you read the proposal, it clearly stated that the community reached out to us because they noticed these high levels of cancer in their community. So that was a big barrier, and I think maybe something New Frontiers needs to communicate to all of our universities, that some of these projects are initiated by the communities and not by the researchers themselves. So.
I think we're going to go in order across. So.
Yeah, so I mean, I actually have a background in physics and then kind of went into biology. And I can say that I think it was actually quite a positive experience, because I think a lot of at least the biological community and the animal behavior community was very welcoming of people from different disciplinary backgrounds. And I've also recently just started collaborating with some social psychologists. And one thing that is been kind of surprising to me is how similar the language of animal behavior and social psychology is. So I think that's something that can often be a challenge in these like interdisciplinary collaborations, and I'm sure even more when you sort of get broader and move across societal sectors. I mainly have experience just within science, but yeah, I think that's something that is important to think about, is like the language that we're using, and sometimes we're saying the same things or using different words to say the same things. But I personally have found that those barriers are possible to cross as long as you, clearly explain what you mean, and with communication, basically. Yeah.
Okay, just to add a little bit, and I want to highlight, like maybe some of speakers already highlight in the previous session. So one of the issues that I encountered, especially during a pandemic, is language is important, and how we communicate. And scientists, my background is chemistry, but when I moved to social scientists, in fact, I still have time to learn to define. And when I talk to civil society, which is sometimes housewives or maybe farmers, I cannot use positive tipping points to talk with them. So I really need to think, like, to get insightful narratives from them, I have to translate this kind of language into their context. So within the chaotic of pandemic, it was quite, really challenging. And also I want to contribute to one of the participants during my research, because one of them actually is a victim of a pandemic, and they passed away, and indeed the narratives are still there.
Yeah, I had the privilege of working cross-disciplinarily, essentially for all my research life, starting from my master thesis and PhD. So I have quite a colorful and wide range of experiences. And let me say first that I find it incredibly enriching to reach out to different other fields and learn from them. So this is my overriding remark. Whatever the costs, the personal gain in terms of intellectual stimulation and creativity are really boundless. Also, you could say in terms of culture, the different fields have different cultures, and it's quite a difference when you enter a physics department or a biology department, and it's kind of nice to be familiar with both. But a little bit more concretely, it's often said that our disciplinary academic environment is putting the dampers on cross-disciplinary research. And by and large, I think that is true. If I go over the very different experiences I had, I would, however, see two different types of cross-disciplinary work, or maybe two ends of a spectrum. The one happened, fortunately for me, very early. When I came from physics, I had all my head full of methods from nonlinear dynamics and stochastic process theory, and I started to interact with a theoretical biologist who had questions, well-defined, precise questions in a language he could explain to me, and I could basically help him solve these questions in a short amount of time, and that was enjoyable for both of us. So well-defined questions and a common language accelerated this productivity of the cross-disciplinary interaction tremendously. And then later, I had other interactions where the questions were not so well defined. Actually, the challenge was to pry out the question, to distill the question from this interdisciplinary dialogue. Also, the language was not common yet, and we had to spend a lot of time developing it. And in my experience, those types of more burdensome or slow cross-disciplinary interactions are better suited for a later part of the career when you're not so much under publication pressure anymore.
Okay, so as the last, they have already put everything nicely, so I don't have much to say, but only one thing if I can add to say is like, so when I started collaboration, at the beginning I thought that I have to entertain my possible future collaborator and, because I wanted to get their attention, then I thought that I can work well. But again, like you guys said, of course we have language barrier, culture barrier, and then it's really difficult to come with. And I just thought that, why do I need to do this? These guys are expert in their field, right? Why don't I just come up and say, hey, this is my problem. Can you solve it? And that was more straightforward. And they said, oh, yeah. Right, so I think you can just keep it simple and then just basically approach to those people who's expert in this field and just show your respect, and then it works. So that's my comment.
I'd like to open it up to questions from the audience. If you have some for a more general question to start for the panelists, so we can go across all of our panelists. Pavel?
So thank you and congratulations again for this fascinating expose of different angles in I think one of the most important items we have, how to involve institutions, humans, behavioral, in this transition which we are heading for. So I have two questions. One is systemic, other one is, Julie, to you. On the systemic one, so basically we have heard here 1+3 different perspectives. I will start with Ulf, who presented to me very well known because I was privileged to work with him for 8 years at YASA before, mathematically modeled systems in which systems are mathematically responsible, they are very much parameterized. And he mentioned that he concluded that, of course, these are agents which don't think, which don't feel, which don't have any rational or any emotional feelings. So he said there is an opening. Then we had 4 examples, right? We had one from Tomoni, showing that actually we can go quite deep into human brain to understand the language. We heard many times from others that language is important in the communication. Then we heard that we know about the meerkat social behavior more than about humans. Actually, this is what I'm saying. When I joined HSSB, I think we understand more of that. Again, going deeper into the brain. Then we heard about the social institutions and humans in terms of the response in Indonesia. So these are real people, right, responding to the real challenge. And then we heard about the very specific issue of the ethnicity and one of the most important challenges, which is cancer. That's a connection as well. Ulf, if you would have a choice to spend all the money of the world with your colleagues over there, how would you incorporate these insights, which are all needed to your models? What would be the way to go ahead? How would you put them together into a little project, big project, to make a real step ahead in what we really need to do?
Yeah, thank you for this question. As you noticed, I pitched my whole presentation on this idea of model-based integration, because I found, well, thanks to my background probably as in theoretical physics, that once you cast a question in a quantitative way with precise variables and parameters, a certain clarity emerges that often remains elusive when one just writes about it in words. But that doesn't mean that this model-based research is the only way, and I recognize that. Actually, I profit a lot from reading interesting philosophical essays about social phenomena that inspire my thinking. So it's complementary, it's not exclusionary, it's complementary. So if you now ask, what do we need for model-based research on communication, coordination, cooperation to move forward? Well, I think there are so many research fronts that it would be arrogant to pick just one. But I mean, since you ask, I would think cognitive biases are so essential, at least for environmental action. Whether people are willing to invest into the next generation, whether they are altruistic towards their peers, is an issue that is still not sufficiently understood, and it sails under the issue of cognitive biases, right? Because cognitive biases are defined by not being maximizing your payoff, so it's actually a derogatory name for a good thing. But understanding that and understanding how maybe some nudging, some context, some environmental feedback can promote pro-social and pro-environmental action, that I think one could investigate in specific stylized experiments. I mean, not numerical experiments, but human experiments, and try to translate that into models and try to translate that into actionable work.
That question of unlimited resources and where would you put your attention if you were not constrained by unlimited resources is one that really cuts across all of these questions. Would one of the rest of you like to take that on?
I mean, I also kind of would throw it back and say, of course, we don't want to invest all in one thing. We've talked about this multiple times, that we need to understand a variety of different interacting processes in order to make progress. One thing that I think has come out a couple of times across different topics is this idea about collective systems and sort of emergent processes and tipping points. I think multiple people mentioned this. And to me, I think this is something that we really need to understand better, especially in human behavioral systems, because we as a species are facing the biggest coordination challenge that we have ever had. And I guess like one thing that we see coming out of even really simple models of collective behavior is that there are a lot of instances in which there's multi-stable states, and also there's memory within these systems as well. So if you just change the rules in one direction and then move it back, you won't get back where you started. So I think on the one hand, these are kind of scary, these ideas of tipping points, but potentially these could also be a bit hopeful, because maybe in these types of complex systems, when you need a very big group level change, it doesn't necessarily mean that you need always a very big individual level change. And so maybe thinking about, are there sort of levers that we can push on in terms of human behavior that might generate feedback loops that would be helpful in tackling some of these larger problems? I think that would be really interesting to explore.
Yeah, I think that leads to the question of tipping points. Are there specific things that you think, with resources available, could push on those tipping points and actually begin to catalyze change?
Is that for me, or? I'm not a social scientist, so well.
No, that was for Cynthia.
I would jump in and say, I think the pandemic taught us a lot. I think many of us thought, especially in the first 4 to 6 months when everyone was locked down and suffering and begging for a vaccine, literally. And then this bizarre phenomenon happened where suddenly it took too long. They weren't trusting scientists. They weren't trusting big pharma. And we ended up with a group that just became strong anti-vaxxers. They don't trust the system. And many of them also felt disenfranchised prior to that. And so I think one of the things that we have to do is to figure out how to get those people that are feeling disenfranchised, and this is Ulf's, hopefully his expertise, we have to figure out how to get that group of really vocal and incredibly powerful people across the world now in every single country to trust scientists again. I think they've lost a lot of trust in scientists. And we have to get, and I think that's the biggest challenge that we have before we can do all of this, but if we still have people that don't trust us, we're going to have that lovely graph that you had where it's not going to move, or it's going to get worse. So.
Okay, I'm going to turn to the audience. I see a hand right there.
Thank you. I have a question to Ulf. You described modeling, different levels of modeling with scales and so on. So I'd like to know do you have some insight into the modeling of collaboration between institutions and individuals, citizens, in the context of policy making, or in the context of taking up innovations, technological innovations? Do you have some insight into how a modeler should characterize the behavior of institutions? Yes, thank you.
Yeah, thank you. That is a very good question that I've also grappled with throughout my research life. So if you want to make an impact on politics, I think the short answer is you have to invest a lot of time and energy and heart into the interactions with the political institutions, with ministry, bureaucracies, with appropriate agencies, and keep pushing for years and years, build relations of trust and gradually expand them. So that is very much along the co-creation and co-development paradigm. And I think that is how it usually works. I have seen this operating at IASA, the National Institute for Applied Systems Analysis, where I worked for 25 years in Austria, together with Pavel, where models of transboundary air pollution ended up, or actually entered the political debate so early that the responsible people in the ministries were very pleased with relying on the data. It was an honest broker role that the scientists could play, and then kept relying on that modeling and kept also funding the expansion of that modeling for 2 or 3 decades now. So that is, I think, the route, very labor intensive, very resource intensive, and very much focused route that I think works. On the other hand, you have occasionally the fashion-driven, I would almost say, leap to the forefront of an issue, and then maybe one is very fortunate to be asked by some politicians or some people in the bureaucracy to help with that issue. So that's sometimes happening, but I think it's far less frequent.
Okay, other questions? We have our spotter. It's kind of hard to see from here because of the light. We got it. Okay,
First, go ahead. Can you hear me okay? Yeah. Thanks to all of you for really informative, inspiring talks. I have a question for Juliet. The statistics you provided on the incidence of breast cancer in West Africa are staggering. And I'm wondering if there's any information about environmental factors that may be contributing to this extremely high rate.
So again, part of the challenge is this trust issue. Mm-hmm. So it's multifaceted and complex, and we don't have enough Black researchers or Black clinicians. And as we sadly know, many of the universities and hospitals in Africa are under-resourced. So that's a huge challenge there. And some of it is as well, there are not as many hospitals in the rural areas or clinics. So the women, when they go to finally decide to go, they are at very advanced stages. So that's why the mortality rate is as high as it is. If they were going a little earlier, it probably would be a little lower. But there's also I would say, in Black communities in the Caribbean and in Canada, there's a stigma as well about cancer. Women and Black communities are more than happy to talk about diabetes, heart and stroke, every disease but cancer. And they will actually, when they're talking about it, they will say, oh, so-and-so has just been diagnosed with the C word. They will not use the word. There's a huge stigma. Yeah. So that's one of the reasons, like, I partner with a cancer support group in Toronto, and we're pretty much doing a roadshow too many times a year. I have to remind them that I have a full-time job. But, we do a roadshow to basically increase awareness about cancer, to tell them what I call Cancer 101, explain what a cell is, what is a gene, what is a protein, because they really don't understand it, and they think it's a curse.
Right.
And so we're starting,
certainly that with mortality rates, but even just the incidence, I think you gave very high statistics, and you also mentioned this location in Canada, in Nova Scotia, where there's a toxic dump. So I was wondering if there's evidence for something like that going on.
In West Africa.
Yeah.
Possibly, as I said, that's what we're, so there's little, but again, there are not that many labs studying this globally. So there's maybe 4 or 5 of us in North America and a few in West Africa, but again, West Africa is highly under-resourced, and so we need more hands on deck to tackle this problem in Africa. Yeah.
Okay, other questions? Yeah, oh, go ahead.
So I was, in the absence of a lot of raised hands, I will take the privilege of saying that when I was watching the meerkats and the marmosets and thinking about the humans, I couldn't help but think about the dancers last night and thinking about how even without one person leading, you can actually coordinate behavior. So I actually wanted to ask Gloria Benedict if after her presentation last night, she asked for scientists to weigh in about some of the fundamental processes that might in fact be underlying the movement of her dancers. And I'm wondering if after this panel there were specific recommendations from dance that you might give our panelists, or if there are specific questions that you have for the panelists. But I think you have to push the button because your mic is not red. There you go.
Well, actually, maybe it's not so much about the dancing itself now, but one aspect that I missed a little bit from the discussion was that, so we talked about, I took my notes, cognitive bias and nudging and so on, and how to change people's behavior. But we didn't yet talk about how to change people's perspective, because I think that would be the prerequisite, and behavior change would be much easier if you change perspective. And so then coming back maybe to yesterday's work that we showed, like in breath, with this overarching story, yeah, that can change people's perspective, and that would also help us to then change the behavior. So maybe that would be something to add.
Thank you. So I think in particular with disparities, changing the story of science is science to there are people disproportionately suffering from specific aspects of medical illness, and we need to change our focus and our narrative. It is very relevant. We have 5 minutes left, so in this last 5 minutes, if you could go across the panel starting in this direction first, and just give us maybe some last thoughts about changing from communication to action, and whether some of the things that you've learned about language or about communication might actually give us suggestions about moving from talking to acting.
Well, that's an interesting question, but at the same time, it's a very difficult question to answer. Like, okay, so I can't answer for this, but what I can tell you what I think about. It's really difficult to deal with that. And then, like I said, we actually, every day, evolution is still going on. So probably we are changing, right? And just we're not seeing because it's going so slow. But at the same time, like I said, we are having more different type of inputs, or like we didn't have cell phone like 50 years ago, right? But now everyone's using cell phone. So neuroscientists actually make a joke, because since everyone's doing this, probably our sensory region for our pointer will be larger. So that kind of thing can happen, and that will also change people's behavior. So I think there's a lot of factors that we should not skip. And then at the same time, like animals, they're not using cell phone, right? So our evolution might be going a little bit in a different way. So I think that's the reason why we have to keep, thinking, and also the researcher have to keep, looking, whatever the system that you're interested in, to really find the fundamental mechanism, and then also whatever cell phone case or AI comes in, and that can actually change that kind of direction. But I think we can't stop this, right? And then we don't know whether it's good or not. At least it's good for the life, but I don't know if it's good for the brain at least, but we need to know what is exactly happening, and I think we are for that.
Yeah, thank you for a very pertinent question. I would like to answer with 3 brief statements. So first, I think we must all guard ourselves against a certain delusion of grandeur, which comes from thinking that our research is so fundamentally important that everybody needs to listen. I think that is rather widespread in the scientific community, and it's totally unrealistic. So I think we must sort of cut the ties with that perspective. And then secondly, the post-rationalization phenomenon that we see in most humans is very strong. So often people have a gut reaction to some issue, and later they collect the rational basis or justification for this gut reaction. And that means that if you enter the interaction with them past that post-rationalization point, it's basically a lost cause. And I think many of the issues have evolved in such a fashion that positions are so deeply entrenched that you can endlessly battle, but it doesn't move the front anymore. And then thirdly, as a little bit of glimmer of hope, in the 90s there was a strong emphasis on chaos control. Maybe some of you remember chaos control, using the complex dynamics of nonlinear systems to nudge them at the right point, in the right direction, in the right magnitude, to enact certain changes down the line that were far bigger than the nudge. So I think once we understand better these complex nonlinear dynamics of social interactions and public debates, maybe that will become possible, and probably means that we have to enter topics which are not yet in the stage of lock-in.
Nice. For me, because I did empirical research, I just want to share my journey in order to ask all of the stakeholders just to stop and listen, so they try to change their perspective, like from no interest to interest to my research. So basically, thanks to the artists that inspired me yesterday, and I learned that language, again, still important, and given us some creativity to our research at least in social science, and then give a creative language, like, the persuasive language to our participants and understanding their position. I think at some point they will stop and listen and give an interest before making some changes.
I thought it was interesting that you asked the question in terms of how to move from communication to action. Whereas at least thinking from a meerkat perspective, we know that these things are intricately linked, right? And often you're communicating and you're acting, and that affects your communication. Maybe just a little metaphor, like in the meerkats, we find that they have these calls that they use to initiate movement and potentially changes in direction. And it turns out if they just make the call, not much happens. If they just move, not much happens. But if they call and move, then that turns the group. Right, so I guess the point would be maybe thinking about how we can both communicate and act at the same time, and how we can possibly generate some feedbacks between communication and action.
I like that.
And I would say that I think as scientists, we all need to learn multiple languages, not like just French, Spanish, whatever, but we need to learn to communicate with the disenfranchised. We also need to learn to communicate with those that are not as well educated, and to communicate our science in what we call a layman's terms, because there is this perception that we're an elite class of people that don't understand the average person. And that's, definitely over the past decade, working with the Black communities in the Caribbean and in Canada, that is pretty much the message loud and clear. They want to trust us, but they are not sure that we genuinely care, and they feel like we speak down to them. And so I think that that's something, if we want to move to action, that's something we should all work on immediately, how to communicate our science in a way that everyone can understand and buy into it, because that will then help us to move collectively faster, I think. Just a simple one to do.
And with that, I think that's the perfect last word for this panel. Thank you very much to all our panelists. Thank you to you for your attention. And now I will turn over the microphone to Pavel, who will, to Guntram, sorry, who will lead us through the last few moments of our symposium.
You can give that to the technician. So, you may have come to notice that we are a speaker short, one of those three that failed to notify us that they are not coming to Paris. So we changed a little bit our program. I asked Pavel, and he's okay with it, and hope you are also okay with it, that we have a short wrap-up by Pavel addressing our symposium, and afterwards we have refreshments and coffee served outside. Okay?
Well, sometimes I wish I would be able to see the world from the meerkat perspective, or a dolphin, or bacteria which is fighting together a virus, all of that. Dear colleagues, I think we have witnessed a very important thing last 3 days. It was a fascinating week by all possible measures which we set to ourselves to measure ourselves after we accomplished those 3 days. As you know, there were 3 major propositions which we, about a year ago, when we met in small circle, put as the objective for this meeting. First, to emphasize again a fundamental role of the discovery science, frontier science, for the progress of humanity. And to put fundamental life science in the broadest context of the world, in all the collaboration with all the disciplines, in the middle of a rather new domain for the fundamental life science, and that is how to use it to build the new bridges with the scientists developing climate models, ocean models, food security scenarios, to better understand the functioning of our Earth system, but mainly to find quicker solutions to the challenges which we have ahead of us. We wanted to build the new bridges between academia, most of them are here, between funding agencies, we had many with us, between the politics and strategic lines, ministers, chief scientists, to build the bridges which will last, which will not end by an event, Paris, June 2023. And I think we were very successful in that. On top of it, we also were able to explain to each other what do we mean when we talk about 21st century being century of biology, of age of biology. Not only we understood the challenge and the fundamental role of the frontier science in that challenge, but also the opportunity and threats of that context. And we also understood how the age of biology, through all the innovation which it will bring, can contribute to the solutions of the environmental sustainability challenge. So I think we could applaud ourselves for this achievement. I am sure that the ministers of different countries, chief scientists of different countries, funders of different countries, will go back and will be fertilized by this atmosphere here about thinking more in the context of out of the silos, thinking more horizontally, thinking more interdisciplinary, thinking more in the fast technical innovation cycle, thinking outside of the classical division between fundamental science, applied science, innovations, thinking in the systems. I think the word of system was used by every one of you in a different context. I would like especially mention the last session, because for me and for many colleagues here, being physicists, mathematicians, system scientists, we knew that technology and the overall progress in technological context is rather far. I think you saw yesterday this example which was given by Naki, showing that we could say that about 50 instruments which use electricity over the last 50 years, we brought together into little thing here, which does all the functions. I use this about 50 times less energy. I mean, this type of examples. But we also know that there is one big unknown in the system. That unknown is connected to the last session here. How you and I, how my ratio, my emotion respond to all of that. I don't know about you, but I don't respond to United Nations Declaration number 333. I read it. I respond to what I feel, what convinced me, what is my language, maybe language of my mother in the future, I don't know, of my grandmother. How humans transform to the future. Humans form the institutions and the agency. So I think the last session was fundamental. It was the start of the new year, I believe. I think this is something where we really could continue very proactively bring together the fundamental communities. Yes, trying to understand how meerkats collaborate and trying to understand how people don't collaborate in this context. So what can we put together? I believe we have a number of outcomes, both on a short and long term. I think the overall outcome is that we were able to increase the awareness of this immense opportunity to build the new bridges. The awareness which I think is taken back by politician ministers, by the funders, and by academia like you. That is lasting one. Second, I think I was witnessing here and also in the corridors by the coffee, new science community is starting to emerge. I saw many conversations between colleagues who never met each other before and who are thinking about how to tackle this opportunity, maybe to propose a project to HSSPO or maybe to some other agency. So I think that process is very important. We witness emergence of a new community here. I think we also see short-term response. I had the opportunity to speak to heads of many agencies who were here in my bilateral conversations. And I know that by this meeting, they were reconfirmed what they have been trying to do. I, for example, would refer to UK, where I think we had an excellent contribution by the head of the UCRI, which is the consortium of all the agencies, which is trying to put this horizontality and this out-of-silo concept into their programming. And I know that other agencies in the room, for example, NSF is still here, is doing the same. I remember when I met first Teresa Good, she is our trustee here. Teresa, are you still here? Yes. We invented together a strange terminology, horizontal biology. Horizontal biology, across all the directorates in NSF. They don't call it like that, but they do similar thing. They call it Convergence Directorate. So I think the funders who do this already got extra inspired. Those who didn't so far, I think they got infected by this idea. This is, I think, the outcome we will see then. I think at the highest level, at the level of science ministers, responsible for the long-term science and innovation vision and strategies, every country has one. Some of them for 10 years, some of them for 6 years, Japan for 6 years. I think they also brought and took back important message to the same effect. Let's have a system view on the way how the future of the basic and applied and innovation cycle will be put together. So we'll be working hard with all of them from the HSSP, but also from all the partners here in the room to actually beat on this idea, because we believe that the system thinking, as many of you put it, needs to be implemented at all the levels. I do expect that these opportunities will become very concrete at different levels. For example, I do expect that the next open call of the HSSPO will see many new proposals, putting the frontier life science in the center of this multidisciplinary challenge which we heard about here. I do expect a number of proposals will go up in UK, in US, wherever in the world with this idea. We are also going to work as HSSP closely with Global Research Council, where we are membering as the only multilateral organization which is able to put money into this cross-disciplinary type of research, trying to convince each other and 100+ funding agencies across the world to take up this challenge concretely. And Alejandro Adem, who left, who is now chairman of the governing board, is fully on board to push this idea across all major funders across the world which are part of it. But at the same time, dear colleagues, we should be realistic. We are still sailing here uncharted waters. We know what happens in our laboratories, in our universities. Many of us are professors here. We know that to change the system, we need to change multi-headed animal. It's not only this part of it. It is incentives, which also were mentioned by you. It is a combination of the career perspective. It is the way how we publish and what kind of opportunities are there to publish. Truly multidisciplinary, horizontal system type of research, all of that. So HSSP will be sticking our head, because our head is there to be chopped off possibly, but we are frontier. We can say the things which other agencies cannot. In the so-called frontier initiative. So what we want to do to follow up from this particular 3 days here. First, as the good academic would do, we would like to write it up. We don't know how yet, but definitely we would like to put a high-level opinion piece, commentary in Science, Nature, other journals, and we will approach you in different capacities, being chairs, being keynote speakers, to help us with that. We also, and this is what I got from you, would like to continue on this by a limited number, small but focused frontier workshops across the world, which would deepen further not only on those 4 ideas which we had here chosen as an examples, health and climate, oceans, food security, and social and cognition behavior. We also would like to go further. For example, what emerged from the conversation here, from the panel on the age of biology, from some of your contributions, was that it probably would be fascinating that HSSP could host, together with the clusters of the innovation institutions based on basic science, frontier talks about how basic science chips into the innovation cycle. And I have an offer already from California cluster, and I have an offer from Singapore cluster, and I have an offer from European cluster to be hosted there. I think this is fascinating what happened here. Also what happened here is, across you, there was a proposal that we should tackle issue of diversity, inclusiveness, and equity in a very specific way. And we are frontier institutions, meaning that those who got the proposals funded with us, you are really the Premier League of the world. You are 4% of the world who gets through it. So, but there is yet another 96% of the world. And what do we do with that? Shall we wait yet another 50 years before frontier science get into those grounds? I don't think so. So there are concrete proposals. One of them is on table from Canada, from United States community, from New Zealand, chief scientist whom I spoke 2 weeks ago, from Australia, and from Norway, to put together first frontier conversation about how the indigenous communities could enter the field of the frontier science, not only as a resource, but as a partner. And we had here one example, which was from Canada, where the shocking number was reminded to us that 80% of antibiotics go to the livestock, and we are having this threat of resistance. And what they are doing there, they are looking back into the indigenous community plans to find the solutions. So there are many ways of that, and we will be stepping up into this ring, using this richness of the membership of HSSPO to put together such a conversation. If I would have a choice, which frontier workshop first, I probably would go Ted and Marina for the social dimension of it. And I think yesterday, Ted, if I'm correct, we kind of agreed that you would host it, right, in Canada? Thank you. So, at the highest levels where we are privileged to operate, you know, HSSPO is a child of G7 decision, and we were very privileged to be on the G7 agenda last May in Japan. We are very proud to say that there is a strong signal that Italian presidency of the G7 next year would take this notion further. And of course, within our sphere of being in this context influential, we would take this idea and work very hard from HSSP to have the declaration next year again, having the science innovation very central as it was in Japan last year. Dear colleagues, let me end on the personal note. Some colleagues here in the room told me, well, I understand finally what you are doing. You are combining your old hobby and new hobby. Well, I do. I do. Because I have been now in Strasbourg for 2 years, and I'm telling to my colleagues and to my trustees that I'm still waking up occasionally, trying to believe that what we can do as HSSP is possible in the current world. In the current world, where we are going back from globalization to individualization, where the world is getting divided and in the blocks again, in the world where international scientific collaboration is there by the virtue of us, but not really supported. I'm very proud that we are the only organization in the world where the members put their resources into one single pot from which we are asked to find best of the best in the world. I believe in it. I believe that model needs to be sustained, not to replace other models. I believe that it deserves a good collaborative construct between the mission-driven science and bottom-up science by curiosity. And I think this particular meeting helped to this notion, because we were able to put the fundamental science not only in the center of the innovation as we know it, but to open up a new frame to show that you doing meerkat experiments or hyena experiments, I think it was as well, and Tomoni doing measurements of the human brain using CT and MRI, that's not an ivory tower only, as people say. That's actually science which can help us to tackle most important challenges of next, not 100, 20, 30, 50 years, which is sustainability transformation. I think that message ministers and the policymakers understood. The basic science is there to bridge, to partner, and to help. I thank you very much. I hope you enjoyed these 3 days enormously as I did, and I hope to see you all in different contexts, in different next steps after these fantastic 3 days in Paris. Thank you so much.